Morning, everyone, and a very warm welcome from the Alberta Diabetes Institute. 100 years ago, the Canadian scientist Banting, Best, Collett and McLeod discovered insulin, and this discovery fundamentally changed a diagnosis from a death sentence to a lifelong treatment for diabetes. Millions upon millions of people globally have benefited from this remarkable discovery in the last 100 years, but we still don't have a cure. Today's symposium celebrates this discovery and highlights the contributions of the scientists involved, including James Bertrand Collett, a U of A professor on sabbatical at the University of Toronto. In the last 100 years, research has not stood still, and the exciting diabetes research landscape in Canada will be discussed. Specifically, the amazing contributions of our own researchers right here at the University of Alberta in the development of the Edmonton Protocol for human islet transplantation and the future of stem cell technologies and stem cell-based transplants will be discussed. A huge thank you to our slate of speakers for making this special event a reality, as well as the efforts of our staff and members of the ADI's trainee working group. I hope you enjoy this symposium, and we should all be extremely proud of our Canadian diabetes researchers of the past, present and future as we strive towards a cure for diabetes. With that, I will pass you over to our hosts, who are members of the ADI's trainee working group, Theo and Stephanie, who will oversee the symposium. Thank you and enjoy.
Thank you, Dr. Light, for the introduction. Good morning, everyone, and thank you for attending. My name is Theo, a graduate student in Dr. Patrick McDonald's lab and chair of the Alberta Diabetes Institute's trainee working group. For today's 100 Years of Insulin event, Stephanie and I will be your hosts and moderators. Please note that questions for each speaker will be answered at the end of their respective talks, and your questions can be submitted at any time using the Q&A feature at the bottom of the Zoom window. Our first speaker is Dr. Alison Lee. Dr. Lee is a historian of science and medicine based in Toronto. Her interest in hormone research originated from studying bovine pituitary glands as an undergraduate summer student in Calgary. She went on to earn her Bachelor's of Science in Biochemistry at McGill and followed up with her Master's and PhD in the History and Philosophy of Science at the University of Toronto. Dr. Lee is the author of the work titled J.B. Collett and the Development of Medical Research in Canada. Here, Dr. Lee goes beyond J.B. Collett's role in isolating insulin and expands on his contributions in shaping medical sciences and laboratory research in Canada. Dr. Lee takes great pleasure in bringing the wonder of science to non-specialist audiences, especially to those who feel that science is not meant for them. The title of Dr. Lee's talk is Extracts and Enterprise, J.B. Collett, Insulin's Forgotten Man. The platform is yours, Dr. Lee.
Thank you so much, Theo, for your wonderful introduction. It's a pleasure to be with you today to celebrate 100 years of insulin. This morning, we'll be traversing a century of history, looking at the challenges as well as the triumphs that researchers have had in trying to understand and treat diabetes. In recent years, of course, as Dr. Light told us, University of Alberta scientists have become known and celebrated around the globe for their contributions to diabetes research. And it's a real honor to be sharing the stage this morning with several of them. What is less well known is that the connection between the U of A and the treatment of diabetes goes back 100 years. And it's my privilege today to share with you the story of these beginnings. I've entitled my talk, J.B. Collett, Insulin's Forgotten Man, but I'm very pleased to say that at the Alberta Diabetes Institute, he is far from forgotten. Collett features on ADI's website at the very beginning of your Our History page. And I'd also like to recognize the work of Mr. Rolly Tipper and the members of the James Bertram Collett Memorial Committee who are working to commemorate Collett in his hometown of Belleville, Ontario. But regrettably, to much of the public, the discovery of insulin is still synonymous with Frederick Banting and Charles Best. Those who know the story better might know that the Nobel Prize for Insulin was awarded to Banting and J.J.R. McLeod. Some might be puzzled to learn that Banting immediately announced that he would share his prize with Best, and that McLeod followed suit by sharing his prize with J.B. Collett. So who was Kollep? What did he actually do? And why did MacLeod feel that his contribution deserved to be acknowledged? This morning, I'd like to answer these questions for you. My talk will take 4 parts. First, we'll look at Kollep's early life and his connection with the University of Alberta. Next, I'll talk about his role in the insulin research in Toronto in 1921 to 22. Third, we'll see how the insulin discovery affected Kollep's career. And finally, we'll end by looking at how the story of the insulin discovery has evolved over the years. James Bertram Kollep was always known as Bert. He was born in 1892 in Belleville, Ontario, the first child of Mahala Vance and James Dennis Kollep. His mother was a school teacher and his father was a florist with a thriving business and a shop on Belleville's main thoroughfare. Bert's lifelong love of the automobile began early, as you can see in this photo. He's driving here with his mother, father, his sister Rita, and his aunt. Bert attended a one-room country schoolhouse and Belleville High School, and he earned money selling vegetables from his grandfather's market garden. But he developed an interest in chemistry and a wish to go to university. So in 1908, at the age of 15, he put on his first pair of long pants to go off to Trinity College at the University of Toronto. He had wanted to study medicine, but because he was too young to go into the program right away, he entered the honors physiology and biochemistry program instead. This rigorous program was designed to create an elite stream of medical students who were trained in scientific research. Kollep graduated at the top of his class, and inspired by the research ideal of his professor, A.B. McCallum, decided to pursue graduate studies. He figured out quickly that McCallum checked in on the lab at 11 or 12 every night, so he made a point to always be there so he could make a good impression on his professor. He soon learned that these quiet hours were also the most productive for lab work, and out of that came a lifelong habit. In his master's research, he found evidence of adrenaline in the nerve cells of the leech, and in his PhD, he studied the secretion of hydrochloric acid in the stomach of various species of mammals, birds, reptiles, and amphibians. He courted a fellow student at Trinity's sister college, St. Hilda's. Ray Vivian Ralph was fun-loving and vibrant, and she was the one who would climb the windows to visit Bert. They were married in 1915 and would have 3 children, Margaret, Barbara, and Jack. Just before he completed his doctorate, Kollep was hired as lecturer in biochemistry in the Department of Physiology at the University of Alberta. The medical school there had been established just 2 years earlier by the visionary president, Henry Marshall Torrey. This picture shows Athabasca Hall, where Kollep had an apartment for several years, and in the background you can see the power plant, which is where the medical school was housed in those early years. When he arrived in Alberta, the boyish-looking Kollep was just 22 years old and not much older than his students. He blushed when they congratulated him on his marriage. Though he was reserved, he could be full of enthusiasm and speak off the cuff at length when there was something that caught his interest. Of course, during this period, the war loomed over everything. Many of the students and faculty enlisted, including the head of Kollep's department, Herbert Mosher, who organized a field ambulance battalion made up of students from the 4 Western universities, and he took them to France. In his absence, Kollep was left carrying the load of all the teaching for the whole department. This left him little time for his beloved research. He had no research assistants and limited resources. And he and the other faculty members really felt a sense of isolation in those early days because few guests came out west and train travel to conferences or to meet with other researchers was very expensive. The library subscribed to a few medical journals, but it took them a long time to circulate. So to deal with this problem, Kollep joined with his fellow faculty members to form a journal reporting club. They gathered regularly to share a meal and present abstracts of recent articles. And I believe there's still a journal reporting club at the U of A that is named in Kollep's honor. The summers gave him a reprieve from the teaching. He and his family enjoyed visits to the research station at Nanaimo, where his shooting, fishing, and clam digging provided him with a variety of research subjects, from starfish, jellyfish, and sea urchins to lampreys and snakes. The end of the war brought new opportunities as well. He spent a stimulating summer in Britain in 1919, which inspired a flurry of 17 papers relating to respiration, adrenaline, and osmotic pressure. Collip, however, felt very hurt when Tory hired another candidate to take over as chair of the Department of Physiology after the tragic death of Mosher in action in France. Collip felt he was the obvious candidate since he had held up the Department of Physiology all through the war years. And perhaps to make up for this, Tory applied for a Rockefeller Foundation traveling fellowship for Collip. So in the spring of 1921, Collip and his young family headed east on a 15-month sabbatical leave. They would begin with a stop at the University of Toronto, and from there, Collip had plans to visit researchers in the United States and Britain. I'm sure he was full of anticipation at this exciting opportunity. What he didn't know was that this would probably be one of the most significant sabbatical leaves ever granted at the University of Alberta. I now turn to the events at Toronto in 1921. First, what was life like for someone with diabetes in 1921? Diabetes had been recognized since antiquity by a patient's unquenchable thirst, fierce hunger, frequent urination, and sugar in the urine. Rapid weight loss would follow. In older people, the onset might be gradual, but in children and young people, death would typically follow within 1 year of diagnosis. No treatment had been found except for a strict starvation diet that might have prolonged life about 2 to 3 years. More than 30 years earlier, researchers had begun to connect diabetes to a malfunction of the pancreas, and by 1901, more specifically to the islet cells of the pancreas. There was no clear consensus about just how the pancreas controlled sugar, but a leading hypothesis was that the pancreas produced a hormone that allowed the body to metabolize sugar. And if that were the case, isolating this hormone might just be the key to treating diabetes. But for more than 2 decades, attempts to isolate such a hormone had failed. Frederick Grant Banting was a 29-year-old surgeon who had served on the battlefields of France during the Great War. After his return home to Canada, he set up a private practice in London, Ontario, but he had very few patients, so he had to supplement his income by teaching at the University of Western Ontario. He didn't have any particular interest or experience dealing with diabetes, but one night after preparing a lecture on carbohydrate metabolism, he woke up at 2 a.m. with an idea. What if those attempts to isolate the anti-diabetic hormone had failed because the digestive enzymes of the pancreas were destroying the hormone? He pulled out his notebook and sketched an idea. Perhaps by tying off the ducts of the pancreas, he might be able to get rid of the cells that made the digestive enzymes, and therefore he might be able to capture the hormone secretion intact. Banting took his idea to J.J.R. MacLeod, professor of physiology at the University of Toronto, who was a respected researcher in carbohydrate metabolism. MacLeod had doubts about Banting, who, after all, was a researcher, had no research experience, and he was a physician who had little more than a textbook knowledge of the field. And what's more, MacLeod was all too aware that there had been many failed attempts to make an anti-diabetic hormone. But in the end, he agreed to support Banting, saying to Banting's chagrin that even negative results would be valuable to science. So in May 1921, Banting sold up his practice and moved to Toronto to begin his research. MacLeod offered him laboratory space in the building shown here, several dogs, and the assistance of Charles Herbert Best, a 22-year-old fourth-year student in the same physiology and biochemistry program that Collip had been in. Best would spend the summer working with Banting before he planned to begin his master's degree and medical studies. So through the summer of 1921, the two young men, advised by MacLeod, worked in a sweltering lab, learning how to carry out the tricky operation of tying off the pancreatic ducts of their dogs, waiting for the Acini cells to atrophy, and then making an extract of the remaining tissue. They then tested this extract on other dogs that they had made diabetic by removing their pancreases. This work was frustrating and slow, with many of their animals dying of infection. But by August, they had succeeded in getting some promising results. They were able to create an extract that caused the reduction of blood sugar in their dogs. And by December, they had made another important discovery. They could make this extract from whole pancreases of cows. So what that meant was that Banting's original idea of tying off the ducts turned out to be completely unnecessary. And what's more, they could now get the starting material in huge quantities from the slaughterhouse. The problem, however, was that this extract was full of toxic impurities and couldn't be used in patients. Earlier in May, Banting had met Kollop in MacLeod's office. Kollop was interested in the project and he offered to help. He was planning to spend the summer at Woods Hole Marine Biological Laboratory near Boston, so he gave Banting his address in case he had any questions. Then, when Kollop returned to Toronto in the autumn, he continued to ask after their work. By December, the research was going so well that Banting urged MacLeod to accept Kollop's offer of help. So Kollop joined the team on December 12th. The events that I described happened over just a few short months in 1921. This year, we are, of course, marking the 100th anniversary of the discovery of insulin, but it really pays to think more carefully about what we mean by using the word discovery. By MacLeod's estimate, there were already some 400 attempts to make an extract of the pancreas, and several of these are particularly noteworthy. Georg Sulzer, an internist in Berlin, Ernest Lyman Scott, a young master's student in physiology at the University of Chicago, Israel Kleiner, a researcher at the Rockefeller Institute in New York, and Nicolae Pulescu, a professor of physiology in Budapest. These and others had already made pancreatic extracts and had shown that they could affect blood sugar and other key signs of diabetes. But when used in humans, these extracts cause terrible side effects such as fever, chills, nausea, and swelling at the site of injection. What this means is that until December 1921, the Toronto team had not done anything that had not already been accomplished years before and published by other researchers. Banting and Best, like these others, had all made an extract that undoubtedly contained the active principle, and they had managed to demonstrate that this product could reduce signs of diabetes. What none had been able to do was to make a product that could be used continuously in human therapy. What took the Toronto researchers beyond this point was what happened during an incredible month, beginning December 12th, when Kollop joined the team. And here, I think Kollop's experiences in Edmonton, in thinking in broad physiological terms and working with rather limited resources, may have shaped the way he approached his work. And he also clearly benefited from having met other researchers and learned new approaches during his sabbatical. Kollop set to work in a systematic fashion. He quickly discovered ways to speed up the process. He found he could use normal rabbits to test the strength of the extract rather than having to use dogs that had been made diabetic. So this obviously gave him a readily available and much less expensive assay. He also used a new, less cumbersome technique of blood glucose analysis that he had picked up from the biochemists at the Nix bench during his summer at Woods Hole. In rapid succession, Kollop made several key contributions. First, he demonstrated that the extract reduced ketones in the urine. Until this point, the Toronto researchers had only measured glucose. Second, he showed that the extract allowed the body to convert glucose into glycogen in the liver. These findings were really important. Banting and Best had shown a drop in blood sugar, but this was not at all conclusive because blood sugar can be affected by other factors such as shock or the presence of foreign proteins. Collip's data provided evidence for the first time that the extract actually restored the ability of the diabetic body to metabolize sugar. At times, Collip noticed that as his batches became more and more potent, his test rabbits became so hungry that they began to eat wood shavings. One day, a rabbit went into convulsions. At first, Collip thought the extract must be toxic, but suddenly he had an idea. He quickly took a blood sample from the rabbit and he set it aside. And then he grabbed some sugar, made up a solution, and injected the animal. The rabbit recovered. An analysis of the blood sample showed that there was almost no glucose in it. Collip had discovered the hypoglycemic reaction. A colleague who was in the lab at the time recalled, "It all looks simple now, but it was the most thinking per square meter per minute that I have seen." For Collip himself, this month felt incredible. In early January, he wrote to Henry Marshall Torrey saying, "The crucial experiment was tried out just before Christmas break, and the results were so striking that even the most skeptical, I think, would be convinced. I have never had such an absolutely satisfactory experience before, namely going in a logical way from point to point into an unexplored field, building absolutely solid structure all the way. We have obtained from the pancreas of animals a mysterious something, which when injected into totally diabetic dogs completely removes all the cardinal signs of the disease. Just at the moment, it is my problem to isolate in a form suitable for human administration the principle which has such wondrous powers, the existence of which many have suspected, but no one has hitherto proved. To be associated in an intimate way with the solution of a problem which for years has resisted all efforts was something I had never anticipated. I only wish that the various papers which will be published on this work were coming from Alberta rather than Toronto." But tensions were growing. Banting began to worry that he was being left out and insisted that they do a clinical trial using a crude extract that he and Best had made. So on January 11th, 1922, Leonard Thompson, a young boy with diabetes, was injected intravenously with Banting and Best's extracts. The results were disappointing. There was no clinical benefit, and what's more, painful abscesses formed. Years later, Banting would acknowledge that their results that day had not even been as good as those of Zulzer in 1908. In the week that followed, Collip threw himself into the purification problem. And on January 19th, while working alone late at night, as usual, using a series of different concentrations of alcohol, he discovered a way to trap the anti-diabetic hormone in a semi-pure form. At that moment, as a colleague put it, Collip actually saw insulin. Collip wrote to Torrey a few days later. The problem seemed almost hopeless, so you can imagine my delight when, about midnight one day last week, I discovered a way to get the active principle free from all the muck with which it appeared to be inseparably bound. Unfortunately, relations among the discoverers became fraught at this point. Banting grew anxious that MacLeod and Collip were taking credit away from him, and an angry confrontation broke out. Collip's extract was tested the following Monday on Leonard Thompson, and this time the results were dramatically different. There were marked effects on sugar and ketones. The patient felt stronger, brighter, and more active. This was the first unambiguously successful clinical use of a pancreatic extract in a person with diabetes. Collip's 4 key contributions during this remarkable month took the Toronto group past what had been achieved by previous investigators. In one short month, Collip had accomplished what had eluded other researchers for decades. Their extract was presented to the world soon after and given the name insulin. The significance of insulin was quickly recognized by the medical community, And in Toronto, banting became the focus of attention as people with diabetes arrived from near and far, desperately seeking this life-saving treatment. MacLeod's oversight was critical. In contrast to the popular image of science as fueled by genius and passion and ambition and ruthless competition, we see here that MacLeod's quieter qualities of caution, conservatism, studiousness, and devotion proved key to the success of the whole venture. He threw the personnel and resources of his department into working out the physiological aspects of insulin, and he coordinated with many clinical partners around the world to evaluate the effects on patients. This provided the crucial supporting evidence needed to present a convincing case to the scientific world. A patent was taken out in the names of the three Canadians, Banting, Best, and Collett, who in turn assigned it to the University of Toronto's Board of Governors for the price of $1. The team collaborated with Connaught Laboratories and Eli Lilly and Company of Indianapolis to work out the complex problems of scaling up the production of insulin for mass manufacture. Many researchers worked on this and in less than a year developed several improved methods for purification. By early 1923, manufacturers were able to produce a standardized, safe, stable, and reliable supply of insulin that was up to 100 times purer than the original version made by Collett. In October 1923, after a breathtakingly short interval, Banting and MacLeod were awarded the Nobel Prize in Physiology or Medicine for the discovery of insulin. And in sharing the prize with Best and Collett, they acknowledged the teamwork that had been the heart of the discovery. In May 1922, the insulin team broke up when Collett left Toronto. And I now turn to the impact of these events on the rest of Collett's career. So what do you do when you are 29 years old and you realize you may very well have made the most significant contribution you're ever likely to make? For some people, it might well ruin them, but for Collett, it would shape the rest of his career for the better. Albertans celebrated Collett's achievement with awards, grants, and gala dinners. At a banquet for 400 at the McDonald Hotel, Collett was greeted with cheers and celebrated as a co-discoverer of insulin. The Alberta legislature voted a resolution expressing the gratitude of the people of Alberta. Collett returned to the university as a full professor and the head of a newly separate department of biochemistry in the splendid, newly opened medical building. The insulin discovery was a watershed in his research life. Before insulin, as we have seen, he had been interested in a wide range of topics in the biochemistry of many species. But after insulin, he would focus exclusively on identifying and isolating hormones of value in the treatment of human disease. Essentially, he would try to repeat the model that he had used so successfully with insulin. Of course, he already had a PhD in biochemistry, but he decided he wanted to go back to study medicine. So he took a medical course right alongside his own students, even though he was one of the department heads. In the first months after his return to Edmonton, before insulin was commercially available, he manufactured insulin in his own lab. An 8-year-old girl from Vulcan was brought out of a diabetic coma with insulin made from Collett's lab. Hers was one of the first handful of these dramatic cases that seemed to observers almost like resurrections from the dead. You'll remember that the patent rights to insulin were controlled by the University of Toronto. The insulin committee there kept half the income for a research fund, but the other half was divided among Banting, Best, and Collett to support their research at whatever university they were at. This was long before there was large-scale government funding of medical science. So this arrangement gave Collett access to what was probably the largest source of ongoing research funding in Canada. And you can see here that in 1925, the department already received $8,000 in insulin royalties. Collett's first major project after his return was trying to find a source of insulin in plants. Initially, there was a lot of excitement about this possibility. He called this product glucokinin, but unfortunately, it turned into a dead end. But he didn't give up, and he soon found his footing again. He typically worked around the clock, often sleeping on a couch in the lab so that he could wake up in the middle of the night to take blood samples from his animals. And the most significant achievement of this period was the isolation of the parathyroid hormone in 1924. In 1928, Kollop was appointed chair of biochemistry at McGill University, and there he headed a large, thriving research team. Members of the department produced research of international significance, with over 200 publications on pituitary and placental hormones, including developing an extract of ACTH for clinical use. Kollop was a talented biochemist, and he loved spending long hours at his still, brewing extracts from huge bubbling vats of glands. The room he was in was filled with the rancid smell of caprylic acid and the deafening roar of a high vacuum pump. Kollop, who his team called the Chief, would run back and forth adjusting this or that mixture, smoking large hand-rolled cigarettes and dropping ashes on his vest. He even got a phone installed by his still so that he could take calls from his stockbroker while he was doing his brewing. Many students and postdoctoral fellows passed through the lab, and they thrived in this atmosphere of enthusiasm and productivity. People from other departments also knew that they could drop by at tea time to join the lab members in their animated discussions. Of course, when Kollop moved to McGill, the insulin royalties followed him. In 1930, Kollop also isolated a substance called emmenin, which is a form of estriol from the placenta. This became really commercially significant because it could be taken by mouth. He collaborated with Ayerst, McKenna, and Harrison, which was then just a small Montreal firm. Ayerst expanded its research and manufacturing operations to make emmenin, and this would lead to Ayerst's development of Premarin. And you may know that Premarin is used in hormone replacement therapy, and it would become one of the top-selling pharmaceutical products in the world by the end of the 20th century. It would also establish Ayerst as a major player in the pharmaceutical world. Ayerst's labels for emmenin and Premarin advertised that they had been standardized in Kollop's lab, and Kollop's department received royalties from both emmenin and Premarin. So by the time Kollop left McGill in 1947, he had brought a total of $750,000 in combined royalties, roughly $10 million in today's terms, more than half of which came from insulin. Kollop as a person was famous for his restless nature and for the speed with which he thought, spoke, and made decisions. He drove so quickly that his students called car trips with the Chief a low form of flying. In comedy skits, they indicated his departure by tossing a handful of gravel against a wall. Banting became reconciled with Kollop in 1925. Here is an exchange of letters which shows Banting expressing his hope that they could bury the past, and Kollop's gracious reply that he regretted the unfortunate misunderstandings of that hectic winter. And here they are meeting at the Calgary Stampede in 1933. In 1938, Banting was asked to form an associate committee on medical research for the National Research Council, and he asked Kollop to join as one of its first members. During the war, Kollop's friends were amazed by how vigorously he took to committee work because he had never had much interest in administrative matters before. In 1941, Banting spent an evening with Kollop in Montreal before setting off on a wartime mission to England. Kollop would be the last friend to see Banting alive. Banting's plane crashed over Newfoundland, killing him and all but one aboard. After Banting's tragic death, Kollop took over his heavy wartime responsibility of overseeing medical research for the armed forces and serving as Canada's medical liaison officer to Washington. After the war, he took a leading role in shaping the coordination and funding of medical research on a federal level. In 1947, Kollop became Dean of Medicine at the University of Ontario, Western Ontario. He enjoyed the challenge of building up its medical school during an era of expansion. He was no longer involved in research by this time. After having been away from the bench during the war, he now found the modern methods of biochemistry somewhat baffling, but he liked to look in at the lab that he had that had been named in his honor. One of the important achievements coming out of this lab was the isolation of the chemotherapy drug vinblastine by his longtime associate Robert Noble and Charles Beer, thanks to the observations of Helena Chauchaska. Ray was a gracious and supportive partner throughout their marriage. She learned to keep a suitcase packed and ready because she never knew when Bert might come home announcing that they were going to leave on a trip the next day. Their eldest daughter settled in Montreal. Their second daughter, Barbara, became a geriatrician in Rome, Georgia. And their son, Jack, was for many years an anesthetist at the Misericordia Hospital in Edmonton. Ray and Bert had 13 grandchildren. Collip never lost touch with his friends and colleagues at Alberta. Here he is reunited with his journal club at a dinner at the McDonald Hotel in 1934. And later, when a plaque was unveiled in his honor here at the medical building in Edmonton in 1959, it was an event attended by dozens of leading Edmontonians. Collip said, "It is the most wonderful thing that has happened to me in my life." Collip died of a stroke at the age of 72, 2 days after arriving home from yet another of his famous cross-country driving trips with his wife, Ray. During his lifetime, he received many honors acknowledging his contributions to science and the leading role he had played in shaping medical research in Canada. And I now finally turn to looking at how the story of the discovery of insulin has evolved over the past century. As the years went by, the discovery grew increasingly associated with Banting and Best, thanks to their efforts and those of their supporters. Soon, any school child would have told you that insulin had been discovered by Banting and Best. Collip, who had left Toronto in 1922, and MacLeod, who returned to Scotland in 1928, were increasingly left out of the narrative. At Toronto, chairs, buildings, and research funds were named after Banting and Best. But for decades, there was no trace of MacLeod, who was one of their first two Nobel laureates. At times, heated controversies would erupt as historians dived back into the original papers and questioned the Banting and Best mythology. Collip always refused to be drawn into any discussion, saying that he had pretty well managed to avoid unpleasant arguments. He said, "Personally, I'm quite happy to let the insulin story stand on the publications by the group who collaborated in 1921 to 22. In 1982, the publication of Michael Bliss's definitive history of the discovery decisively changed the narrative because of his compelling argument that insulin was the result of Banting, Best, Collip, and MacLeod. In the decades since, he and others have argued that Collip probably should have been included in the awarding of the Nobel Prize. If you haven't had a chance to read this book, or if you want to read it again, I'm very happy to tell you that a special centenary edition is coming from the University of Toronto Press. I was honored to provide the foreword to this edition. This book is not only important, but highly readable, and I heartily recommend it to anyone who has an interest in this topic. The other investigators I mentioned, Zulzer, Scott, Kleiner, and Pelescu, have also had their champions, though the uncovering of Pelescu's association with the fascist and viciously anti-Semitic Iron Guard in Romania has drained away interest in celebrating him as a hero of science. In their stories, there were all the elements needed for scientific breakthrough: insight, ingenuity, dedication and persistence, biochemical expertise, painstaking experimentation, clinical studies, and pharmaceutical industry collaboration. These researchers had the right elements. They just didn't have all of them together and at the right time. It was at Toronto that expertise and tenacity, laboratory and clinical resources, and the collaboration of the pharmaceutical industry were all brought together exactly when they were needed and in peacetime. And this coming together made all the difference. The heart of the story, however, is not the disputes over credit, but the impact of insulin on the lives of people with diabetes. Before insulin, diabetes was a death sentence. After insulin, it no longer was. The coming of insulin was an undoubted good, but not an unalloyed one. In the century since, people with diabetes and their health providers have had to learn how to manage a complex chronic disease. Our next speakers will be telling us more about that history. In July 1922, Teddy Ryder was 5 years old and only 22 pounds, one of the so-called living skeletons brought to Toronto for treatment. He was among the first to receive insulin from Banting. Later that year, he was able to write to Banting, "I wish you would come to see me. I am a fat boy now, and I feel fine. I can climb a tree." Teddy Ryder would have a chance to grow up, thanks to insulin. In 1990, Ryder returned to Toronto when the largest auditorium in the medical faculty was renamed in honor of MacLeod, and a permanent exhibit reflecting the contributions of all 4 discoverers was installed. Ryder would be the first person to live for 70 years on insulin. Last month, Historica Canada released a Heritage Minute on the discovery of insulin. I was honored to serve as one of the historical consultants for this piece, and I came to appreciate the huge effort that went into distilling this complex story into 1 minute of screen time. We were very conscious of wanting to fight the Banting and Best mythology, knowing that this minute of film might have a big impact on the way future generations of students remember the insulin discovery. After months of working through many versions of the script, our producer asked us to circle back to the heart of the matter. What was the one message we needed to convey about insulin? Was it the Nobel Prize, the teamwork of the 4 scientists? Was it that they sold their patent for $1? I'll show you the video now, and I hope you will feel that we got it right.
Leonard Thompson, 13 years old, diabetes mellitus, 65 pounds.
Starve the child to let them live? The treatment's as cruel as the disease. It's a death sentence.
Dr. Banting.
This could be it.
He's the first to receive this trial.
But will it save him?
It's not pure enough.
So we try again. And again. And again.
Before the discovery of insulin, diabetes was a death sentence. Banting, Best, Collip, and MacLeod's breakthrough has saved millions of lives. Leonard Thompson's was the first.
As for the question of what we mean by the discovery of insulin, I'd like to leave the last word to Collip himself. The Muttart name will be very well known to you in Alberta for the family's many contributions to philanthropy. In 1959, Gladys Muttart wrote to Collip of her plans to donate a Wurlitzer organ for the chapel of the University Hospital. She wrote, "Being a diabetic for over 26 years, we know that if it had not been for your work with insulin, I would not be alive and enjoying good health today. Therefore, we would be delighted if you would allow us to have this organ installed in your honor with a suitable plaque engraved as follows. In recognition of the contribution of James Bertrand Collip to the discovery of insulin." Collip replied saying that he was deeply touched, but suggested one change to the wording of the 3rd line. Instead of the discovery of insulin, he substituted development of insulin therapy for diabetes. He said, "Although I have been guilty of using the word discovery on occasion myself, generally I do not like it. Most so-called discoveries represent simply the last but important step in a long series of previous steps representing contributions of many others through the years in the scientist's search for truth." In 1923, at a glittering banquet celebrating the Nobel Prize, there were many tributes and speeches, but I think Llewellyn Barker of Johns Hopkins summed it up best when he declared that there is in insulin glory enough for all. So today, a century later, I believe we should think more expansively about who and what contributed to the coming of insulin, knowing that there is indeed glory enough for all. Thank you.
Thank you, Dr. Lee, for that captivating recollection of Column's research efforts. I've learned so much. We do have time to answer a few questions before moving on to the next speaker. We have one from Cheryl Howell. Alison, do you believe the recognition of Banting and Best as the discoverers of insulin is correct, or do you advocate for this to be changed to recognize the others involved?
I absolutely feel that it's very important to recognize the 4 key discoverers, Banting, Best, Collittman, McLeod, and I was very glad to hear that in many of the discussions today and elsewhere during this centenary of the discovery of insulin, that the 4 names are being recognized.
Perfect. Thank you, Dr. Lee. As we move on to the next talk, I would like to let everyone know that we are actively reporting this event through the Alberta Diabetes account on Twitter, which has been taken over by our trainee working group colleague, Saloni. Feel free to post about this event using the hashtag Insulin100. Our second speaker is Krista Lamb. Krista Lamb is a Toronto-based writer, communications professional, and podcast producer and host. Her specialty is helping translate complex medical, scientific, and health-related topics into interesting and understandable stories for diverse mediums. Krista Lamb is the producer and host of the Diabetes Canada podcast, which won the Canadian Podcast Award for Outstanding Health and Fitness Series in 2020. Further, she also hosts the podcast From Beta Cells to Bicycles for the BC Diabetes Research Network, as well as hosting the podcast Actions on Diabetes with Diabetes Action Canada. She recently published her first book titled Beyond Banting, From Insulin to Islet Transplants, Decoding Canada's Diabetes Research Superstars. She is particularly passionate about the role of research in establishing new treatments and one day an eventual cure for diabetes. The title of Krista Lamb's talk is Beyond Banting, From Insulin to Islet Transplants, Canada's Ongoing Contribution to Diabetes Research. With that, the platform is yours.
Thank you. I'm looking really forward to talking to you guys about this. I will warn you, my presentation is very different than probably all of the other ones that you're going to see today. I really focus a lot on what is happening now and on telling stories. I am not an academic. I am a journalist by training. And one of the things I loved was the opportunity to be able to share some of these stories in a journalistic fashion. I will start with my disclosures because I do work for a number of nonprofit organizations. I always like to start with that. However, we won't really be talking about any of them today. I will also do a land acknowledgement. I wanted to start by acknowledging the land on which I'm speaking. For thousands of years, Toronto has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and I am grateful to have the opportunity to work on this land. So you've already heard the introduction about me, so I won't go into a lot of detail. But the thing that is important to know before I start my talk is that I am one of the luckiest people I know because I get to do what I love for a living. And that is tell the stories of people who research diabetes and the stories of people who are really bringing the science to life. So every day I get to tell different and unique stories in different and unique ways. And I've come to realize that science communication is so important to this field and to making sure that other people understand outside the academic and scientific circles why the work that's being done is so important. I started a lot of this when I did the Diabetes Canada podcast. We're in season 5 now, and we've been able to tell the stories of many researchers from across Canada. But what I realized as I was telling those stories on the podcast, which is about 15-20 minutes about an individual research project, that there was so much more I wanted to talk about. And so that inspired me to write my first book. It is called Beyond Banting, From Insulin to Islet Transplants, Decoding Canada's Diabetes Research Superstars. As you can tell from the title, I am a very big fan of the work that you guys are doing. I think it's really important to tell these stories. So why? And that's a big question that I often grapple with, and I get asked this a lot. I think that scientists focus a lot on getting published. I think all of you know that that's the end goal for many of your projects. You want to be published in a journal, and that is an exceptionally important goal. However, I like to try and tell people that that's not the end goal, that there is so much more that can happen after that publication or even before that publication that can have real impact. And that can have impact on things like funding, but it can also have impact on the people in the communities, because the more people who understand why this research matters, the more likely they are to support things like funding, and the more likely they are to realize that the work being done truly, truly matters. I want people to hear these stories and walk away from me saying, I didn't know that. And that is something that makes me so incredibly happy when I'm able to inspire someone with one of the stories of the work that you guys are really doing, and to get them to really care about that. Now, I will not be going into a lot about the discovery of insulin. I think we just had a wonderful overview of that, but I like to steal from a popular meme and look at how it started. I've had the pleasure this year of speaking at a number of the 100th anniversary celebration events across Canada. And what has happened in those 100 years is incredibly remarkable. And we should be very grateful that we had these four men come together to create this discovery. However, I'm a big fan of how it's going, and how it's going is pretty awesome. I think that sometimes we don't acknowledge enough about where we've been and how we've got there and the people that are doing work right now. So why Canadian diabetes research? Funny story, people ask me this a lot because you will have a hard time finding another person who is as enthusiastic about Canadian diabetes research as me. And I think there's a lot of reasons for that. When I first went to Dr. Dan Drucker and asked him about this project, he acknowledged that we do punch above our weight. When you go to an international conference, when you sit down and look at who the people who are putting out papers are, considering the size of our country, our diabetes community is really, really contributing significantly. However, the general public, and I think Dr. Lee touched on this a little bit in her talk, they don't know very much beyond Banting. They will often tell you that, oh yeah, we have a really great history of diabetes research in this country, but the only thing they will tell you about is Banting. And I think that that is something that we really need to change. For example, I was here in Edmonton pre-COVID to interview some of the people involved in the Edmonton protocol for the book. And I was having a wonderful discussion with a woman in a store about the reasons I was there. And she'd never heard of the Edmonton protocol despite living in Edmonton. And I thought that's so disappointing, because I think it's such an important thing and I wish more people knew about it. I also think that our young scientists really benefit from understanding the successes and seeing how far we have come. I know from a lot of the trainees that I've become friends with or that I talk with regularly that it can be really frustrating to be sitting in your lab and, the middle of the night, or getting up to check your samples or looking at cells and not getting the results that you want. And so I wanted to tell them some stories that I hoped would inspire them as much as I am inspired by this work. So first things first, I could not tell every one of the stories that I wanted to. There are so many people and so many things that have contributed to the history of Canadian diabetes research. I would have been writing forever. And so I had to really look at what are the stories that I thought would capture people who weren't necessarily from this academic community. What are the things that I thought they would want to know and find interesting? And so I chose stories that I found really interesting as someone who isn't a scientist and isn't an academic, but also stories that I felt really resonated. And it's been so interesting to me as I've toured with the book across Canada to different universities, the stories that the researchers themselves have pulled out of the book that they found inspiring, because they've often surprised me. Now I'm going to tell you about a few of the people that are featured in the book. I'm going to do this because I think it's important that we get a sense of some of what's happening across Canada and some of the incredible work that has been done. For example, I hope all of you are familiar with this gentleman. This is Dr. Daniel J. Drucker from the Lunenfeld-Tannenbaum Research Institute in Toronto. He recently was a co-winner of a Gairdner Award, or a baby Nobel, as we like to call it. And we were so thrilled for him because Dan has done so much in terms of bringing forward GLP-1 agonists and DPP-4 inhibitors, which are changing the lives of people living with type 2 diabetes. However, he will be the first person to tell you that he did not do this in a vacuum. In fact, he talks a lot about the collaborative process that it takes for these discoveries to happen. And so that was something I really wanted to highlight, because I know there are researchers from UBC and from across Canada that have all had a hand in moving these projects forward. However, when it came to the chapter itself, I also wanted to tell a story that hadn't been told about Dan, because he's had a lot of people writing about him over the years. And one of the things I found most interesting when he and I talked was that he spoke a lot about how, when he decided to work on this project, he had the end goal of getting a therapy into clinics. And so he worked through that process, and he had a roadmap for that. He went from the discovery phase to the clinical trials phase. He went and got pharma support. The drugs were now available on the market. All of the things that you think will lead to success. However, what he realized when he got to that point was that his therapies were expensive and they weren't easily available to the people who needed them. So he has done a lot of work then to really try and focus on how do we get people these drugs? How do we get people to access this? And so I speak in the book to Dr. Christopher McCabe from Alberta, who does a lot of work with CADIT about how drugs are priced and how drug availability is managed. And I thought it was such an interesting story, because I think for many of you who are looking for that next step in your discovery, you don't think a lot about the process. And Dan acknowledged that he hadn't thought much about that process until he got there, of how we get these discoveries to the people who really can benefit from them. Now, don't worry, I will not be telling you about the Edmonton Protocol today, because I feel like this audience is pretty familiar with that story. And I know that the speakers coming up are going to have a lot to say about it. However, just like with Dr. Daniel Drucker, I could not have written this book if I did not have the access to the team from the Edmonton Protocol, who were so kind to invite me to come and meet with them in Alberta and to be able to sit with them for hours and talk about the work they've done. So I am incredibly grateful that they gave me as much time and as much access as they did. But what was really interesting to me when I looked back at the Edmonton Protocol was what I call the cascade effect of this. The funding for a project like this is enormous, and the need is ongoing. And so to have the Alberta Diabetes Foundation formed because of, the Islet program, that was a really important thing for me to note. I also thought it was really wonderful how the Alberta Diabetes Institute was built to continue on this legacy and to continue on this work. And then Islet Core, which is also, moving projects forward by taking donor tissue and ensuring that nothing is wasted and that things that are donated to the program can go on to help with other discoveries in this area. It was really fantastic to think that it's not just about the Edmonton Protocol, it's about so many things. The other thing I wanted to talk about, and I won't talk about the story of Bob Teskey, it is in the book and it is incredibly important, I think, to this. But what I do want to talk about is, again and again, I met people while I was doing my research who were patients who said yes when they were asked to be part of a research study. Now, many of us who are in the research area know how necessary that is. But I think it's really important that we also talk about how brave that is, to be able to say yes to an untested treatment, to be able to say yes when you don't know what the outcome will be, and to be part of that history and to be part of that. And I know that being able to share Bob's story was really important because of that. And you will be seeing some of these faces later today. I always like to say that Canada has some amazing stem cell superstars, and talking about the contribution of Canadians to stem cell science is something I could not have written the book without. Stem cells were, of course, discovered in Toronto. If you're in Toronto, you can go to the Mars building and you can have your photo taken with Till and McCulloch's statues. And, we are rightly proud of that contribution, but I think we should be just as proud of what is happening in in labs today, where people like Dr. James Shapiro, Dr. Tiimothy Kieffer, and Dr. Cristina Nostro are doing wonderful work in trying to use stem cells in different ways for therapies to diabetes. Now, one of the things that I won't, like I said, go into Dr. Shapiro or Dr. Kieffer because they will be speaking soon. But I did want to talk a little bit about the other two people who are pictured here. Dr. Cristina Nostro, I loved her story because she has gone on to become one of Canada's, if not the world's, most recognized stem cell scientists in the work that she's doing to develop beta cells in the lab. However, she talked in the book about getting to that point in her career where she had to decide what she was going to do, and really just packing up 2 suitcases and going across the world from her home in Italy to work with Dr. Gordon Keller. And I know that many of you who have done your postdocs or are doing your postdocs have done the same thing. And I think it's really important that we acknowledge the importance of that and how fantastic it is that people can take these steps and go towards finding this work. I think it's just incredible bravery and tenacity. And I really felt it was important to recognize that we were lucky to have some of these international researchers who have come to Canada to do their work, but also that it was a really big leap of faith for them to do that. I also like to tell the story of Dr. Derek van der Kooi. Dr. van der Kooi, who is in the corner, is not someone who does a lot of diabetes research, despite the fact that he himself lives with type 1 diabetes. For Dr. van der Kooi, he's really looked at neuronal stem cells. He's looked a lot at brain stem cells and eye stem cells. However, it was in his lab that they discovered neurons in the pancreas, which could have a really big impact on how we look at the physiology and how we look at potential cures or treatments. Now, the reason I tell this story is because they did this completely by mistake or by accident, depending on how you look at it. They had been doing other studies and other work, and they noticed that they were seeing these neurons in the pancreas. And so his trainees went to him and said, "We're seeing this, it's really weird." And he was like, "Did we contaminate the samples?" And that was their first guess, was that they had made a mistake and contaminated the samples, because obviously they work with a lot of neurons. And so they went back and did it again and again and again, and they were able to get the same result. And so I love the idea that some of these things can be a happy accident. We cannot talk about diabetes research without talking about the researchers who live with the condition that they're studying. Some of you might be familiar with the recent project that Dr. Michael Riddell and Dr. Patricia Brubaker have been doing, where they have put together this concise list of diabetes researchers who live with type 1. And it was incredible to see this spread across my social media as people were so excited to be included in this list. And we were able to see the number of researchers who are now out there doing research while living with type 1. And I will tell you that the groups that I have had the privilege of working with have been so inspiring, but they also taught me things that I don't think that we talk about enough. For example, when you live with a condition like diabetes, which is a 24/7 condition, it can also be really challenging to have that as your work. And I spoke with Dr. Desi Sarieva, who was the first trainee in Dr. Michael Riddell's lab, who also had type 1. She's now at Stanford, but she spent her PhD training with Dr. Riddell, and he sat her down when she came to him with the idea of working in his lab. And she said the one thing that he mentioned was, he said, "You are more than capable of taking on this role. You are more than capable of doing this research. But the one thing I want you to think about is that when you live with this every single day, you're thinking about diabetes all the time. Do you also want that to be your work?" And we are very lucky that both she and Dr. Riddell said yes to that, and they're both very happy that they said yes to that. But it was a consideration that I don't think I'd ever taken into my context when I was talking to them until they said that, because they always seem so happy about the roles that they play. And I mean, when you look at Mike Riddell, who studies diabetes and exercise and is a world leader in that field, he lives with type 1 diabetes. He has a child with type 1 diabetes. Most of his trainees have type 1 diabetes. And so this is his life and his work. And that is a pretty spectacular thing. And when I talked to Dr. Bruce Perkins, who studies complications in type 1 diabetes, he in particular was very passionate about his work because he had seen so much work being done towards therapies and cures, but very little being done towards complications, which for someone living with type 1 like himself was really important to him. And so he had gone in, and he's known for doing things like, trying to figure out ways that we can use SGLTIs to better treat type 1 diabetes without having the DKA that we know is prevalent in some cases. So he does all of this work, but he always does it with the lens of someone who is living with the condition. And I talked to him a lot about the pull that he often feels because he has this enormous waiting list as a clinician scientist of people who want to be his patient, of people who want to see him because they think maybe he has some kind of special knowledge as a person with type 1. And while he disputes that, because he says, he doesn't think he does work any differently than other endocrinologists, there's always that pull of could he be doing more? And so I thought that was a fascinating thing to look at when we look at our type 1 diabetes researchers who are integral to moving forward because they understand this condition better than any of us, but who really have a remarkable challenge there. I'm also going to speak a little bit about the work of Dr. Heather Dean. Many of you will be familiar with Dr. Dean and the work of the DREAM Diabetes Group in Manitoba. Now, there is an excellent book called Diagnosing the Legacy, which is all about this project, and I would highly recommend it. I only got to write a chapter, but I could see immediately how an entire book could come out of this. So Dr. Heather Dean, as many of you know, began to see the symptoms of type 2 diabetes in Indigenous youth from northern Manitoba communities in the 1980s. Now, the thing was, Dr. Dean was a very young scientist. She was very new to this field. She was a young female doctor who was new to the field. So as you can imagine, in the 1980s, her work was not always welcomed. And she faced a lot of people who kind of poo-pooed the idea that this was happening, who said, oh, you're just misdiagnosing type 1 diabetes. This isn't something that's a problem. And she disagreed. And we're very lucky that Dr. Dean had the tenacity to continue to do this work, no matter how many people told her that she must be wrong. And it was when she was called to the Centers for Disease Control in the US and asked to start working with some of the researchers from the US who were seeing type 2 diabetes in youth from marginalized communities that she really started to feel like she was getting the acceptance of the scientific community. And I think that's a really important reminder of how we need to sometimes stop and listen to people who might be saying things that don't entirely jive with what we thought was the case. Dr. Dean was also one of the first people who really believed in the integration of the communities that she was studying. The woman in the pink coat in the bottom is Bertha Flett, and she is considered an enormous part of this project, even though she was not a researcher. She was part of the community, and she really helped Dr. Dean to integrate because Heather Dean did not want to come into Indigenous communities and say, you have a problem, I'm going to figure out how to fix it. She wanted to be part of the community and to talk to the community and to work out solutions together. And that's the ethos that continues to be part of the DREAM Diabetes program. I talk a lot about how they have taken those steps and continued to include them and how important it has been in the success of the programs. I also had the opportunity when I was talking to them to learn a lot about how they look at interdisciplinary research. So the blonde woman in the top corner and the woman in the red, that's Dr. Christine Doucette and Dr. Brandy Wicklow. Now, Dr. Doucette does basic science. She's looking at cells in a dish. She's looking at the physiology and studying those things. Dr. Wicklow has inherited the cohort that Dr. Dean had because those first children who had type 2 diabetes in those communities went on to have their own children, and many of them now have their own children. So Dr. Wicklow has been able to study those. However, they work together, which I think is really, really important. And by working together, Dr. Doucette isn't just in her lab, she's in the clinic, she's meeting the people whose cells she's sometimes studying. She's able to talk to the community and to look at the barriers and to understand them as people, as well as have this overarching view of their physiology. And Dr. Wicklow in turn gets to have the ability to work with Dr. Doucette. And so I think these are important things that are starting to happen more and more across Canada, and I'm so happy to see that. And in the blue in the top corner is what I like to say is the person who has changed my life, because I interviewed Dr. Taylor Morisseau for the book, and she is one of the most extraordinary Indigenous scholars that I've ever met, and she taught me so much about how we need to include the experience of Indigenous peoples in research about them. She brought to the table so many things that I had never considered. And because of that, I think she is making the research program in Manitoba so much better, as are the other Indigenous scholars that are part of that program. Now, I've told you a few of the stories about the book, or from the book, and now I'm going to tell you a few other stories. And I'm going to tell you these stories because I think it's important to tell you some of these things in the way that they sparked my imagination, because these are other stories that have come out of these scientific breakthroughs and scientific innovations, but also just general research presentations. So one of the things that I like to tell people about is when I saw Dr. Christine Doucette speak at the BC Diabetes Research Network's Diabetes Day a few years ago. She was the keynote, and she was talking about her work looking at the physiology of traditional versus colonial diets on the human body. And I mean, that's a really interesting topic, but I was worried when I sat down that I wouldn't be able to understand what she was talking about, because it is such a really granular topic when you're looking at this from a basic science lens. However, she started her presentation by talking about what Manitoba and what Winnipeg would have looked like 100 years ago. So this isn't the photo, but it's a similar one. And it really struck me that it's just over one lifetime ago. The land that people were living on and the food that they were eating was so incredibly different, and how that had never really resonated with me. And so it reminded me, and it's something that I talk to scientists about all the time when I'm teaching them some of the techniques to tell their stories. Is there something in your presentation that can spark an idea without the data, or that can take the data and bring it to life for someone who might not understand the data? And this was a great example of that, because Christine was speaking to a room full of scientists, but she told such a wonderful story. This is Dr. Kaveri Dasgupta, who I had the pleasure of chronicling in the book. And she is someone who I think about a lot when we think about how we share our science stories. Dr. Dasgupta has been profiled internationally. The media has done so many stories. When I was doing the research, I was like, wow, you are in the media a lot. And I think one of the key reasons for that is because even though she does work in very different areas, she's a clinician scientist who works in gestational diabetes, type 2 diabetes, and type 1 diabetes. She really looks at everything through the lens of a family problem. Diabetes is a family problem. So no matter what type of diabetes you yourself have, or if you are just a family member of someone with diabetes, it's something that everybody in the family is dealing with, and that has impacts on everyone in the family. And her work has been particularly interesting in that she was one of the first people to look at, we know women with gestational diabetes have a higher risk of type 2 diabetes. We know that their children have a higher risk of type 2 diabetes, but she wanted to know about the spouse. And what she found was that, yes, the spouse does have a higher risk. And a lot of times people hadn't thought about that because there was no genetic connection. And so her work, again, lots of headlines about this because people found it really interesting, but it all relates back to the work she does, trying to figure out how to make things easier for families, and then she can work on interventions to try and improve things. You may be familiar with Dr. David Campbell. He is from the University of Calgary, and I tell his story a lot because it was one of those things that resonated so incredibly deeply with me. Dr. Campbell studies diabetes and homelessness. And we have a lot of misconceptions about this topic. I know I did before I was involved in his work. And he decided he wanted to really spend some time, and he put together a group of people who were experiencing diabetes and homelessness or housing insecurity. And he spent weeks, months with them talking about their experiences. So it wasn't looking at it through the lens of, this is what I think the problem is. He wanted to talk to them about what they saw as the problems. And there were some interesting problems. One of the women I spoke to for this project, she talked about how she had been beaten up and her metformin was stolen. That's not a problem that I have to deal with. And it was just so jarring to me how little we consider the actual realities of a life on the street. And so when he completed this project, Dr. Campbell could have written a research paper. In fact, he did, and it's excellent. I highly recommend it. But it was also important for him to figure out how to share the stories in another way. And so he asked the people in his group what they suggested. And they suggested photography. And from that, the Home Sweet Homeless Project was born. And if you were lucky enough to see it when it was touring before COVID, I hope that it moved you as much as it did me. I remember looking at photos and thinking, my gosh, I had never once considered this. I had one woman, her photo was of what she was given to eat at a homeless shelter. It was cookies and orange slices, and every part of me wanted to say, you should not be eating that because that would be not a good thing for your type 2 diabetes. But if I had no choice, what would I do? And I don't think if I had just read the paper, I would have been as moved if I hadn't had the chance to see her words on those photos and to see those photos. Another gentleman talked about the decision he had to make between buying pizza pops for $5 and a salad, because the salad would only feed him for a day and it wouldn't fill him up, but the pizza pops he could eat for a week. And these were never things that I thought about in terms of diabetes, but by telling the stories in this innovative way, I found it so much more powerful. And I'm going to come full circle with the idea that it can be about more than just the publication. This is Dr. Ahmed Haider. I was able to chronicle his work on the artificial pancreas, which he's doing at McGill University. He's doing wonderful things in the group at McGill in that field. However, part of his research brought him to Camp Carawanis, which is in Quebec. And he spent weeks with these campers over 2 summers, getting to know them, getting to study them, but really enjoying his time at the camp and being part of the community. And what he realized while he was there was that there was an issue that the medical staff were dealing with. He basically realized that if the campers are all in separate tents, and the tents are quite spread out. And so for the medical staff, they would go from camp to camp to tent all through the evening. And they wanted to make sure that no one was having any kind of medical situation with their diabetes or any kind of situation at all. But they never knew who was going to be having an issue. So they could be on one side of the camp and a camper on the other side is having a low, and they have no way of knowing about that. And Ahmed looked at this problem and he thought, I could probably fix this. And so he worked with his students to come up with a technology that the camp staff could have in the medical tent that could read the CGMs and could see in real time if anyone was having an issue. So they would still go from tent to tent to tent, but if someone had a low, they would get an alert in the medical tent, so they would know to go to that tent. And he and I had not been talking about this particular project. I had asked him what he was most proud of. And that was the story he told me. And he said, there's no academic reason for him to do this. He's not going to write a paper about it. It's a local camp with a small number of campers. And if he didn't do it, nobody probably would. But when he presented it to the nursing staff, he said some of them had tears in their eyes because they had worried about this for years working at this camp, and they had struggled with it for years. And with his training as an engineer, he could apply something that could really make a difference for the diabetes community in his home area and these kids that had supported his work over these last 2 summers. And that meant a lot to him. And it really reminded me again that we often think of the publication as that ending step. But being part of the diabetes community, being able to give back to the diabetes community, these things can be so incredibly important. I will also take a minute to talk about the next generation. I always joke that I already need to write another book because I have had the opportunity to see firsthand how incredible the up and coming research community is. We have so many researchers, and I talked about Dr. Taylor Morrisseau earlier from Manitoba and the work that she's doing in Indigenous communities. Dr. Ananya Banerjee, who is just below her, is a researcher I've spent a lot of time working with. Dr. Banerjee is looking at diabetes in the South Asian community. Similarly, Dr. Calvin Key, who is just below her, is working on those sorts of questions in the Chinese community. And I was really excited about this research because I had worked at Diabetes Canada for many years. And over that time, I had been told again and again and again that people from South Asian, Caribbean, Chinese communities are at higher risk for type 2 diabetes. And then people would ask the obvious next question, why? And I had never got a satisfactory answer. So when I started talking to Calvin and to Ananya, and they were telling me about their work, I was so excited that people were really digging into this problem because we need to find these answers. We cannot continue to tell people that they are at risk for something without being able to explain why. And Ananya's work in particular was very interesting to me because I had a lot of friends who were South Asian growing up, including one of my closest friends. And so I asked her a little bit about, this is not a heterogeneous group. This is a huge swath of people from very different circumstances. And she talked to me about the fact that we often don't look at the situations from which people come. So she had looked at the Sri Lankan population in Ontario compared to the Indian population in Ontario. And while both had an increased risk of type 2 diabetes compared to the median, what she found was that if someone had come from Sri Lanka, escaping the civil war there, their rates of diabetes were even higher, giving credence to the idea that there's much more at play here than genetics, and that stress and circumstances can really play an important role. And I think for young people who are perhaps of South Asian descent, who are growing up in this community being told that they are at higher risk, but not understanding why, these are the sorts of things that will be very important to them. That's another reason she has started the South Asian Diabetes Youth Group, which is helping South Asian youth in Peel, which is a part of Ontario that has a very high South Asian population. They put out an incredible documentary recently called Beyond the Body that looks at the experience of young Canadians from South Asian backgrounds and their families. And it talks a lot about some of the misconceptions and the stereotypes and the things that we need to get beyond if we're going to be able to successfully change the trajectory in terms of type 2 diabetes risk for communities. And again, similar, Dr. Key is doing work and has worked in Hong Kong and has worked around the world in looking at the fact that the Chinese Canadian community is not heterogeneous. It is not one community. There are so many different facets and experiences. And so it was fascinating to know that this work was being done. Similarly, Dr. Ruth Najobi, who is beside Calvin there, is doing so much work at the University of Laval in regard to the barriers that people with diabetes from different ethnocultural backgrounds face. And I think these, again, are things that we're not necessarily talking about enough, but can have a major impact on the uptake in diabetes resources. So she's done quite a bit of work around retinopathy screening, for example, and making sure that people have access. Dr. Anne-Sophie Brazo is working with Dr. Remy Rabassa-Laret at the University de Montréal, and they are working on the BETTER study about hypoglycemia and how to prevent it, because this is something that they saw again and again and again becoming a problem within the community. these are all interesting and exciting projects. And I will note the woman at the top corner, Priya Irawima, who is in Tiimothy Kieffer's lab and is an incredible inspiration to me, because the minute I met her, I was blown away by her enthusiasm for this work. She works in stem cell research with Dr. Kieffer, and she has such a passion for science communication. And I think that if I impart anything on you as I close off this talk, it's that the future generation of scientists will benefit so much from being able to tell their stories. And I think that as we see where that work is going, we will start to realize how necessary it is for them to be able to share these amazing research stories. And so again, my book is called Beyond Banting. I want to thank everyone who helped me with this book. So many of the people who are in attendance today helped me in some way, whether it be by doing interviews or talking to me about your projects, or just by championing the book and helping me get to where it is. And so I really am so appreciative of that. I do a lot of discussion about science and science communication, in particular in the diabetes field, on Twitter. So please do feel free to follow me there. I am also happy to talk to and connect with any young scientists or trainees or people who might have questions on LinkedIn or through my email. I always like to end my talks with this wonderful sort of array of pictures from the before times when I could actually speak to people and hang out with the diabetes community. And they have been really wonderfully receptive to this project. And so I am very grateful for that. And now if anyone has any questions, I am more than happy to take those.
Perfect. Thank you for that great talk, Krista, especially the aspect where you point out it's important to not just tell someone they're high risk, but to help them understand why they're high risk and to further explore that. We do have time for a few questions. One of them is from Cheryl Howell. Krista, you focus on telling the stories of researchers. Are you familiar with the story of Islet transplant programs by a participant, One Step Up from a Lab Rat by Donna Marcelson? Do you plan on writing further on this topic?
I haven't heard of that one, but I definitely want to read it now because I did have the opportunity, like I mentioned, to talk to some of the Islet transplant recipients, and it was wonderful. So I definitely would love to read that. I think right now my career is in telling these stories. I am lucky enough to get to talk to people all the time. And so I definitely think that I will be doing more stories about diabetes research and about the future research projects. So if anyone has any ideas, I'm always open to them because I'm always looking for new stories to tell for different projects.
Perfect. Cheryl also wants to know where she can purchase your book from.
Oh, well, you can get my book anywhere that you buy books, really. A lot of people purchased it on Amazon. If you don't like Amazon, my publisher will sell you a copy that is unrelated to Amazon. I am published by Rocks Mills Press. But if you go onto my website, kristalam.com, you can find it all through. There's a question about libraries I just saw. Libraries are challenging, more challenging than I thought when I decided to write a book. So it is available in some libraries, but not all libraries. If you would like it to get it through your library, please let them know, because they often make the decisions about what books to bring in based on requests. So again, it may be in your library, it is in quite a few, but not all libraries. So the things you learn when you write a book.
We do have a couple of more general questions that are available on the Q&A. So if panelists could press the Q&A button at the bottom of your Zoom, you'd see them. I think those two questions are more broad and can be answered by anyone by typing their answers in. I do have a question for you, Krista. So when it comes to communication from a scientific perspective, so do you have any advice for newer graduate students and postdoctoral fellows with regards to how they communicate their research and where they can learn to improve that?
Absolutely. I just did a talk recently for the University of Calgary research residents. And one of the things I was talking to them about is, I decided to do podcasts. I decided to write a book. You might not feel comfortable with any of those things, and that's completely okay. But there are so many options. You can look at the work of, Dr. Samantha Yamin, who is Science Sam. She's sharing amazing research via Twitter and Facebook and TikTok. Her TikTok about the coronavirus pandemic has been so impactful in terms of getting information to people who need it in the space where they need it. And so I always say, what are you good at? What kind of communication do you like? Do you love TikTok? Do you like YouTube videos? Figuring out the way that you want to tell your science stories can really be a personal thing. And you might find that what you do can translate really well to a different audience. Because as Sam always says, we often talk about how people aren't paying attention to the science stories, but are we telling them in the places where people need to see them? Or are we just telling them in our circles? So I always find that really interesting. And if you haven't seen Science Sam, I highly recommend checking out her science communication work.
Perfect. Thank you very much, Krista. Cheryl is excited to go ask a library about your book.
Well, I appreciate it.
We will now move on to our third speaker, who is Dr. Raymond Rajotte. Dr. Rajotte is Professor Emeritus of Surgery and Medicine and the founding director of the Islet Transplantation Group at the University of Alberta. Additionally, he is also the founding scientific director of the Alberta Diabetes Institute. After moving to Edmonton to train as an X-ray technologist in 1965, Dr. Rajotte has gone on to have an exceptionally successful career in diabetes research. With well over 225 peer-reviewed publications, he has established himself as a pioneer and expert in islet transplantation. Since 1986, Dr. Rajotte has mentored over 50 graduate students, numerous undergraduate students, and just over 25 postdoctoral fellows. A testament to his strength as a mentor is that many of his students were involved in the original Edmonton Protocol and now serve as leaders in Alberta's and Canada's medical and research community. Dr. Rajotte is highly decorated. Of note, he was listed in Alberta Venture magazine as one of the 50 greatest Albertans of the last 100 years. He was also the recipient of the Alberta Order of Excellence in 2006 and inducted as a member of the Order of Canada in 2007. The title of Dr. Rajotte's talk is From Bench to Bedside, The Road to the Edmonton Protocol. With that, the platform is yours, Dr. Rajotte.
Thank you. Thank you very much, and I hope you can hear me. I'm going to close this top here. There. So what I would like to share with you is a journey that started in roughly 1964 that allowed us to carry out Canada's first islet transplant, and then on to the Edmonton Protocol and to the World-Class Diabetes Institute. And I really want to acknowledge all the many surgical fellows, graduate students, technicians, as well as administrative staff that allowed us to complete this journey and allowed us to take our research from bench to the bedside to the standard of care for diabetics to the world-class Alberta Diabetes Institute. And it was the contribution of so many people that have made this all possible. Some of these individuals, and you might not recognize them, have been with us for a long time. This is James Lyons that has been with us for 25 years. And we have Deb Dixon has still with us after 30 years. Greg Corbett started off as a technician in 1985 and is still working and is a full professor in the Department of Surgery and is still with us after 36 years. And Norm Kniedemann has been with us for 40 years and he's retiring this summer in July. And last but not least, Pauline started off as working with me when she was 18 and still involved in the Alberta Diabetes Institute. Well, as you heard earlier, Collop was a key figure in the discovery of insulin, and he went to Toronto and he, as you heard earlier, he was the co-discovery of insulin. After he then returned to Edmonton, and he decided to go to medical school, and he graduated from the medical faculty in 1926. After he got his MD degree, he did some research at the U of A, and this is the McEachran lab right here that he did his research. And he was there for a couple of years, and as you heard earlier this morning, he then went to McGill to head up the Department of Biochemistry. About 1959, Walter Mackenzie, who was the dean at the time, decided he wanted to build a research institute. And so he approached Ernest Manning, who was premier at the time, and got the money. And in 1961, the Surgical Medical Research Institute was built, and the director at the time answered directly to the dean, starting off with Walter Mackenzie all the way to Tom Mary. Now, the Surgical Medical Research Institute had about 25,000 square foot of space. It had core labs, surgical ORs, biochemistry, histology, photography, but it also housed a lot of space for researchers. Believe it or not, at one time there was 25 full-time paid technicians at the SMRI. So whenever anybody wanted to do research in the Institute, they were given 2 or 3 technicians to work on a project at no cost. Well, that's where I started my career in 1964, actually in a bathroom. And we built the Human Islet Isolation Lab in the SMRI that allowed us to carry out Canada's first islet transplant, as well as provide the islets for the Edmonton protocol. Pretty well all the individuals that were involved and are involved with islet transplantation, as you can see on the screen here, did their graduate work at the Surgical Medical Research Institute. Even after they were completed and got academic appointments, a lot of them actually also did their research in SMRI. Also, the Human Islet Isolation Lab is where we train people from all around the world how to isolate islets. Well, I started off my career when I started off as an engineer, working with George Bonder, who, as someone I had met as a surgeon at the Emeritus General Hospital, I was an X-ray technician. He was interested in gastric secretion, and so I started to work with him, and I developed a perfusion device to keep the stomach alive using fluorocarbon. Well, fluorocarbon was a compound invented by the 3M company, that in large part for using as an insulator and transformer, but in actual fact had the remarkable property that it carried a lot of oxygen. So I used that as the blood substitute. After I completed my engineering degree, I was very fortunate that John Dossiter moved to Edmonton. And to me, John is one of Canada's greatest scientists. After his lecture, I told him what I was doing, and he's a nephrologist by training, and he said, "Ray, do you think you could cryopreserve the islets?" I must confess at the time, I didn't even know what the word meant, but I said, sure, why not? So I developed a device to slowly cool the kidneys to sub-zero temperatures, then using the fluorocarbon. And back then, you couldn't go out and buy a Toshiba microwave. You had to build your own microwave, and that's my supervisor, co-supervisor Jeff Voss from engineering. And we tried to freeze the kidneys. We had some success, but a lot of the kidneys didn't work long term. Well, that was right around the time that Paul Lacey and Ballinger reported that they could go in and they could isolate islets from one animal, transplant them into the liver, and they were able to correct diabetes. So I said, well, maybe this small multicellular structure might be easier to cryopreserve. I was very fortunate in 1974, the first islet transplant meeting was held in Minnesota, and I had the good fortune that I was able to attend it. It was hosted by Ballinger and Lacey, and also other people that I met that were surgical fellows at the time were Dave Sutherland, David Sharp, and Danny Pipplers, who became dear friends and colleagues as time passed. After the meeting, I returned back to Edmonton, learned how to isolate islets, and then went ahead and developed the cryopreservation technique, adding the cryoprotection to protect the islets from intercellular ice crystal formation, used slow cooling, and then went in, plunged them to liquid nitrogen temperatures, did rapid thawing. And using this approach, I was able to show that the frozen thawed islets were able to normalize the animal post-transplant. I decided then to do graduate work in the US, going initially to Peter Mazur in Oak Ridge, Tennessee, who's an expert in cryobiology, going then to Minnesota to spend some time with Orion Hagerty and David Sutherland, going to St. Louis with Paul Lacey and David Sharp, and then going to UCLA to Yoko Mullen. Back then, as an engineer, it was very difficult to get funding to do postdoctoral training. And I was very fortunate that Tom Williams, who was head of surgery, had a very good working relation with the Eminent Civic Employees Charitable Assistance Fund that had been supporting the Department of Surgery since 1941. He approached Toby Clark at the time and asked him if they would consider supporting Rajotte to do postdoctoral training in the US, and they did. So then I went to the US, starting off with Oak Ridge, Tennessee, Minnesota, but probably my most productive time was when I went to St. Louis at Barnes Hospital. And it was an islet factory. David was running the lab. He had 3 or 4 technicians, and any given day, they would lay 25 to 30 animals on the table. They could isolate all these islets, and in one day, they could transplant 10 to 15 animals. So in a very short period of time, we're able to do a lot of experiments, further optimizing the cryopreservation of islets. I then went to UCLA to spend some time with Yoko Mullen and Joshua Brown. And what Joshua Brown has shown is that he could take the fetal pancreas, cryopreserve it, and transplant it beneath the kidney capsule, and they were able to normalize the animal post-transplant. But another important study is they showed that hepatic secretion of insulin was important for normal physiological glucose control. I then returned back to Edmonton and was very fortunate to get a cross appointment between the Department of Surgery and Medicine. George Mullner had just joined the University of Alberta from the Mayo Clinic. And again, I had no money for my research, so I approached the Eminent Civic Employees Charitable Assistance Fund, and they gave me $10,000 for research. And think about it, that back then, with that $10,000, I was able to run my lab, hire a technician, and pay for all the animal experiments. And they continued to support my research from 1977 to 2007. Now, after I came back, my lab was actually just below where Collip had done his research on the ground floor. And that's where I set up my islet isolation lab and my cryopreservation techniques. And the first project I did when I came back to Edmonton is I showed that I could cryopreserve the islets in Edmonton, put them on a plane in a special container, ship them to St. Louis, and there we would take the islets. David Sharp in St. Louis would take these islets that were the fresh islets isolated in St. Louis and the cryopreserved islets that were frozen in Edmonton and shipped to St. Louis. And you can see if you look at the plasma glucose versus time post-transplant, there's really no difference in the two groups of animals. And then at which point you remove the graft and the animal immediately becomes diabetic. Probably the best thing that happened though was having an appointment in the Department of Surgery. The Department of Surgery had its own graduate program, and you could get an MSc or PhD. And surgeons are in training were encouraged during their surgical training to do graduate work. And so they would come out of their surgical training program and they would spend 2 or 3 years in a lab with a scientist and get an MSc or PhD. This program was also open to basic science. So pretty well all my graduate students that I had the good fortune of supervising went through this program. My first 2 students were in 1981, was Garth Warnick, and then Norm Kniedemann in 1982. That was also at a time when the Alberta Heritage Foundation for Medical Research was established, and there was a real interest to create, if you will, surgeon scientists. So the way I built up the islet transplant group, after Garth had finished his training, He was interested in research. He was interested in working in Edmonton. So I arranged to send him to Oxford to work with Peter Morris and Catherine Fields, after which time he then came back to Edmonton. Norm Kniedemann sent him, he wanted to do a transplantation fellowship, so he went to St. Louis and did a transplantation fellowship in Barnes Hospital, but he worked in the lab of David Sharp and Paul Lacey. Greg Corbett, who did a master's degree under me, after he wanted to do a PhD, so he went to Brussels to work with Danny Pipeliers and got his PhD, and we're fortunate he was able to come back to Edmonton. Jonathan Lakey, we sent him to Indianapolis to work with John Chrysler, and last but not least, Gina Rayotte in 1999 went to Denver to learn transplant immunology. And the first two to come back to Edmonton were Garth in '85 and Norm in '86. Now, the techniques that we used to isolate rat islets, when we tried a similar technique in the larger mammal pancreas or the dog pancreas, it didn't work. We were dealing with a much larger mass of tissue. And in retrospect, we were using tissue choppers and shakers, etc. So what we ended up with is pancreatic microfragments. The question is where do we transplant this large volume of tissue? We couldn't use the kidney capsule. There wasn't enough room. The liver was problematic. You ended up with portal hypertension. So the site that worked pretty well in our hands is we would go in and we would embolize these pancreatic fragments back up into the spleen. And the advantage there is this also gave portal drainage. As reported, that this was an important site to transplant by UCLA, Joshua Brown and Yoka Mullen. And if you compare plasma glucosis versus time post-transplant, you can see transplanting this large volume of tissue in the spleen. The dogs, after a couple of weeks, became normal glycemic and remained normal glycemic as an autograft long period of time, for almost 2.5 to 3 years post-transplant. When Mark Evans came into the lab as a surgical fellow, he showed that he could also cryopreserve the tissue, and it worked very well. Well, the question, as we were starting to think we wanted to transplant humans, we felt that this large volume of tissue of pancreatic fragments would be a major problem. We couldn't transplant it into the spleen. Anatomically, the human spleen is quite different than the dog spleen. So we felt that we had to somehow get a smaller volume of tissue of pure islets to embolize it up into the liver. An important paper by Hiraguchi and Mero suggested that maybe what you should do is to deliver the collagenase via the pancreatic duct. So what we did is various stages of evolution, we developed a perfusion device in which you cannulated the pancreas in 3 or 4 different spots. And using this, you were able to control the pressure, the temperature, and the flow rate at 4 degrees or 37 degrees. And this is perfusing the gland with collagenase. And what this does is you're delivering the collagenase by the pancreatic duct, you're destroying the acidosis here, but at the same time, you're cleaving the islets from its collagen matrix. Following this step, we then tried to purify the tissue using shakers and stainless steel sieves and a cascade of filters. But finally, the method that we settled on was outlined in this slide here. Following the perfusion step, we would then just go in and tease the gland apart with forceps. We then went through what we call a trituration step, in which we just went through different gradations of needle. We went through a 14 gauge needle, a 16 gauge needle. Then we had to purify this tissue, and we used Ficol density gradient. And finally, we ended up with a fairly large volume of highly purified islets. Initially, it was very difficult to identify the islets. You can see it here. The islet was sort of a blue tinge, but we took this preparation and we transplanted it again into our dogs, back onto the spleen. And you can see post-transplant, these pure islets were able to normalize the animal, at which point we went in, removed the gland, and the dog became diabetic. An important paper, though, that came out of the Miami group is that you could add dithazone to the prep, and this stained the islets red. And so this made it much easier for us to quantify the number of islets we were transplanting. And this is when the islet equivalent was, if you will, was invented. And what an islet equivalent is, we went in our preparation, we had made a special chart, and we would actually, with a graticule in the eyepiece, we would size the islets. And so we measured the volume of the islets. So for example, this islet, say, is 150 micrometers in diameter. This islet is, say, 250 micrometers in diameter. So this islet here has about 6 times the beta cell mass of this one. And so what we were able to do is, islet equivalent is an islet of 150 micrometers in diameter. And this was adopted by many labs around the world. So now when we transplant it and we do a dose response, we say, this is the number of islets transplanted per kilogram body weight of 150 micrometers in diameter. So the dose response we carried out is outlined in this paper here, slide here. We embolized the islets up into the portal circulation, and you can see when we transplanted 5,000 to 6,000 islets per kilogram body weight, all the animals became normal glycemic. And so these are islets of this size here. Mark Evans, when he came to the lab, also showed that comparing fresh islets with cryopreserved, they worked roughly the same. And then Mark Cottrell, when he was in the lab as a surgical fellow, he compared autografts with allografts on cyclosporin. And what he found when we did an allograft, it took more islets. It took around 7,000 to 8,000 islets per kilogram body weight. So encouraged by these large animal experiments, we felt we were ready to start transplant humans. And we were fortunate the Alberta Heritage Foundation for Medical Research gave us an establishment to set up the Human Islet Isolation Lab. And then Garth approached the HOPE program, the organ procurement program, and said, could we get pancreases for research? Initially, our pancreases weren't ideal in that we were the low organ on the totem pole, for example. So the heart was first, the kidneys were next, and finally we got the pancreas, but a lot of time with a lot of warm ischemia. So they weren't ideal pancreases. However, we were very fortunate when Norm came back and he started a liver program, he became our champion to procure the pancreas for us, and we ended up with much better organs. And so what we did, we isolated them, as you heard earlier, using the perfusion step, going through all these steps, and finally we were able to end up with a large number of highly purified human islets. Initially, what we did, though, is we cryopreserved them and established a low temperature bank of islets, during which time we measured the viability using perifusion or transplantation in naloxone diabetic nude mice. Also back then in '88, we had no money to run the lab. And so I approached various granting agencies such as the MRC, the CDA, and the JDRF, and they said, no, this is strictly a research, and we can't support that type of research. I was lucky that Don Obrowski, who was chairman of the JDRF local chapter, was also in the national office. And he went to New York to plead my case, and they said, no, the money raised in Alberta has to go to New York. So Don Obrowski said, well, let's start our own foundation. Let's start the Alberta Foundation for Diabetic Research. And Joanne Langner became the executive director for 20 years. And they supported the Islet Isolation Lab from 1988 right up to 2000. Now, I was very fortunate to have incredible dedicated group of technicians and research fellows, etc. That the pancreas always came at night, usually on weekends. And so the technicians had their regular day job, but when we got a pancreas, they would go in and set up, work all night, and then go back to their day job the next day. Very fortunate to have these dedicated group of people. So the first pancreas, first organs, humans we were going to transplant was a diabetic patient that needed a kidney transplant. So what we did is They got the kidneys from the organ donor. We got the pancreas. We isolated the islets, and we were able to carry out Canada's first islet transplant. And this looks at James Connor's results post-transplant. This is his insulin requirement. This is looking at C-peptide, or insulin secretion, if you will. And we had a pretty good response post-transplant to about 15 weeks post-transplant, and then we started to lose islet function. But at no time did he become insulin independent. Our second patient, Kevin, the same type of thing. Had a pretty good response for 6 weeks post-transplant. Then we started to lose islet function. And again, at no time did he become insulin independent. And this is using the cyclosporine A with steroids. And we knew from some studies, including our studies and others, that that wasn't the best drugs to use. In fact, it affected insulin secretion. And what Mark Cottrell had shown is when you're doing an allograft with cyclosporine, that we needed more islets. So then we decided in our third and subsequent patient, we thought, well, let's increase the number of islets to 10,000 islets per kilogram body weight, combining freshly isolated islets with cryopreserved islets. So they got the kidney from the organ donor. We got the pancreas from that same donor, isolated the islets. However, this time we supplemented the fresh graft with cryopreserved islets. And when we did this, you can look at hemoglobin A1C, glucose, and C-peptide, but you can see here, just looking at our glucose, at roughly a month post-transplant, we were able to stop her insulin therapy. And this patient remained insulin dependent for almost 2.5 years post-transplant, which was very encouraging. We did other patients using the same strategy, using a combination of fresh and cryopreserved. Number 4, number 5 here. Both these patients didn't become insulin independent, but they did have long-term islet function. In fact, Greta out to 10 years, and number 5 out to 6 years. So even though they didn't achieve insulin independence, they were able to control their diabetes much better than if they wouldn't have got a transplant. And so around that time, these are the centers that were involved with clinical islet transplantation, and about 200 transplants were carried out, but only about 14% of the patients remained insulin independent at 12 months post-transplant. So because of time, I'll mainly concentrate now on the innovations in islet isolation that allowed clinical trials to move forward in 1999. We continued to improve the way we perfuse the islets and deliver the collagenase. Camilla Ricordi introduced the Ricordi chamber as a way to improve islet isolation. Now, when we were dealing with the pancreas, to purify the tissue was quite labor intensive. We needed 50 to 100 test tubes. And it was the Miami group as well as the Leicester group that introduced the concept that you could use probe 2991 to purify the tissue. And that was a real advantage for us. Also, the collagenase that we used back then in 1989 was very impure. And in fact, and there was a lot of lot-to-lot variation. So we did a lot of work with the Boehringer Mannheim, as did the Miami group, Camilla Ricordi, and we helped the Boehringer Mannheim folk develop a better collagenase for human use, and it was called Liberase. So the procedure that evolved for 1999 is outlined in this slide here. Once we got the pancreas, we immediately started to perfuse it with collagenase. We used the Liberase. Following this step, we would then place it in the Ricordi chamber. And we start circulating the collagenase till about every 2 or 3 minutes, we take a sample. And once we start seeing a large number of highly free islets, we go through a dilution step because we have to get the islets out of the collagenase in this chamber here. And so what we do is we add Hank's balanced salt solution, and we collect it here, collect the islets over the gamish of tissue over here. Then we take this tissue and we purify it on the COBE 2991. And finally, we're able to go from this volume down to this volume of highly purified islets. Now, we're very fortunate in 1995 that James decided to come to Edmonton to do a transplantation fellowship with Norm, as well as learn how to use his term, cryopreserve those damn islets. And so, but he also did a PhD in the lab. Now, we knew that the drugs that we were using back in '89 were toxic to the islet, so what, especially the steroids and the cyclosporin. And so what James did when he came to the lab, his research, he did a PhD as well as a transplantation fellowship. He started to look at different drugs that would be better for the islet function. Very important, got rid of the corticosteroids, and he used the anti-IL-2 receptor as for the induction therapy. And this actually had a 10-week induction period. He then looked at FK506 with rapamycin. Eddie Ryan, one of the team members, felt that we were justified in doing a solitary islet transplant in a brittle diabetic. It was also at a time that James became the clinical director of the islet transplant program, taken over from Garth Warnock. This is the procedure we used, and we transplanted, James transplanted 7 patients. And he had made up in his mind that we would do the first transplant. And because we had a larger induction window of 10 weeks here with Xenopax, he felt that we would give him a second transplant or continue to transplant him until they produced insulin independent. And all the patients got a second transplant during this window of the 10 weeks. One got a third transplant, but more important, all the 7 patients became insulin independent and remained independent for 12 months post-transplant. And as you know, this was then published in the New England Journal of Medicine. And this improved clinical success from 14% to 100% of the patients off insulin. And this really set the standard worldwide for clinical islet transplantation. Well, even before the paper was published, the NIH wanted to develop an immune tolerance network. They were trying to decide who was going to take the lead, and Jeff Bluestone became the leader. There was also an islet subgroup that James and I attended, and they were trying to decide in their own mind, what kind of study could we do for immune tolerance for islets? James presented the work on our 7 patients, and so Aldo Rizzini said, well, why don't we do a multi-center trial using the Edmonton protocol? We approached Bluestone, and he said, write up the proposal. So we invited the Miami group, we invited Bernard Herring from Minnesota and Dave Harlan from Washington to come to Edmonton in December of 1999, and it was minus 40 degrees when they came. And over the weekend, James took the lead, wrote up the proposal to include half a dozen centers, presented it back to Bluestone, and they funded the multi-center trial. And that was at the time, roughly, when we decided to move the islet isolation lab from the Surgical Medical Research Institute over to College Plaza, in large part because the medical building was old, it was in need of repair, asbestos in the ceiling, leaking pipes, etc., and a few cockroaches laying around. So that's when we moved the islet isolation lab over to College Plaza. Initially, Jonathan Lacey oversaw it, but then for the last 20 years, we've had, they've been over in College Plaza, and it's been probably one of the best islet isolation labs in the world. Well, around 2000, the university came to me and they said, we want to take over the Surgical Medical Research Institute. I said to them, and Lauren Terrell had got money to build HRF East and HRF West, but there was no money to fit up the research building. I reminded the administration that back in 2000, about 80% of the work was islet related, including the Islet Isolation Lab. So we had a meeting with Lauren and Stu Hamilton and those involved with the islets, and we said, should we get a CFI grant to build an Islet Institute, or should we build a Diabetes Institute? And we decided, no, let's build a Diabetes Institute. Lauren, the dean at the time, said, who's going to take the lead? Ray, fine. Not realizing what fine meant to me. So the first thing I did a survey of the amount of research that's going on, diabetes research that was going on campus, and there was a lot of activity. And so I thought, well, it would be nice to put all these researchers under one building. And it would cover nutrition, it would cover basic islet transplantation, it would cover exercise, etc. And so what I suggested is all these individuals would then go into the Diabetes Institute. And the Surgical Medical Research Institute would move over to HMRC. And it would become a surgical learning center for robotic surgery and microvascular surgery. At that time, so I had a meeting with the dean at the time, Tom Mary, and I suggested to him, I said, because all the scientists were going to go over into the ADI, what we should do is the 3 administrative staff, we should send them over to the ADI under the scientific director of Peter Light. Then the Surgical Medical Research Institute and the director that always answered directly to the dean, the 3 positions should come to the Department of Surgery, and the director in Rajotte should answer directly to the head of surgery. And they liked the idea, and that's what was done. And it became a state-of-the-art surgical learning center. So we wrote up our CFI grant. It included 5 faculties. It included SMRI, as well as it included the Alberta Diabetes Foundation. Remember, when the new Edmonton protocol became the standard of care and the province paid for the procedure, the Alberta Diabetes Foundation were looking for a new project. And so they decided to give us $10 million towards the new building. And a result for that, they got a space in the Alberta Diabetes Institute. And there was a lot of planning, and from hole in the ground to the move in 2007, it almost took 7 years of planning. And I was very fortunate that John Elliott and Greg Corbett were key in helping us plan the building, as well as Greg Olson, that was responsible for looking after all the equipment and getting multiple quotes for the project. So just looking at one lab, the fifth floor, back then, PI was allowed 750 square foot of space, senior scientists 1,000. And so we put the PIs in the outside. And to get more space in the building, what we built is core labs. So for example, we built a histology core lab that Lynette looks after, but Greg Corbett oversaw the lab. We also built a cellular immunology lab that Colin Anderson looked after. We built a small animal islet core lab, as well as we put a comprehensive tissue bank for low temperature, as well as experimental islet isolation in large animals. And that's now been taken over by Patrick McDonald, and I believe he calls it the Islet Core. And James Lyons actually looks after isolating the islets from the experimental pancreases. And I believe, talking to James recently, he's now done 400 pancreases from research pancreases, which is an incredible asset for the Alberta Diabetes Institute. The lab was opened in 2007 by Ed Stelmach, and we had enough money roughly to complete 70% of the building. Doug Horner was the Minister of Advanced Education at the time, and every year he would give money to the university for completing the projects, but the university always used the money for other projects. we're very fortunate. Tim Schultz was one of our Islet recipients, and at function, I mentioned to him that, we're not getting any money from the province to complete the building. And so the next thing we know, next year, we got 95 million to complete the ADI. So it's not what you know, it's who you know that sometimes makes a difference. And then Greg Corbett in 2014 got another CFI, $26 million to build a GMP to process stem cells and xenografts. So it's been an interesting journey, and it's been an interesting journey for 50 years. And truly, our success has been because of the contribution of so many people, research fellows, etc., that have had the good fortune of working with and supervising. I've been very fortunate to work very closely with the Alberta Diabetes Institute and the Alberta Diabetes Foundation. And these are just some of the individuals that have been involved with the Alberta Diabetes Foundation that have chaired the board. We've had some incredible philanthropic donors. Don Abrowski gave us $1 million. Don Hamilton, because his 3 kids have grandchildren have diabetes, he gave us $1 million. The Allard family, Peter and Chuck Allard and Kathy Rusin, gave a chair in honor of their father, who was a surgeon. And we also got Ernest Wertman $1 million, in large part because his wife was treated by BEST. And we have other very generous philanthropic donors. So I'm optimistic. In the next 50 years, I truly think that we'll have a cure for type 1 and diabetes, type 1 and type 2 diabetes. And in large part, we have incredible, outstanding group of scientists working within the ADI. Cellular therapy, I think we're in good hands with James, Greg, Gina, and working on stem cells and xenografts, Colin Anderson, autoimmunity, Andrew Pepper working on devices, and then Pat McDonald working on working with the core lab. And we have very unique within the facility, the GMP facility to process stem cells, and then the core lab. And then we have, so there's going to be changes within the ADI. Peter has been at the helm for the last 11 years, and we're in the process of looking for a new scientific director. We have a new scientific director for the Alberta Diabetes Foundation. Lee is now taken over as the scientific director, just joined us about a month ago. And I think the ADI is in very good hands. The surgical medical research is now a hole in the ground. However, it'll live on in its new facility with Gina acting as the scientific director, answering directly to the chair of surgery, David Williams. And it's a state-of-the-art surgical learning center. So I honestly think and believe that in the next 50 years, there'll be an insulin-free world. Thank you very much.
Thank you for that great talk, Dr. Rajotte. I recently joined the ADI in 2019, and I've always been fascinated at the diversity of research and fields that are here. And it was great to hear the history of how the Institute and the foundation came together and how it is now. There is an interesting question from Cheryl Howell. It's an anecdote followed by a question.
Okay.
Firstly, she likes to thank you for your work and that the islets that you extracted in your basement laboratory were housed in her liver. And she's eternally grateful for that, having won the Islet lottery with her first transfusion or first infusion. She mentions she recently saw some genetic research that compared the efficacy of medications. And I think she's talking about using genetic profiling as a method to predict how well a treatment can work. Do you know of any research regarding a person's genetic profile with regards to anti-rejection meds required in the Edmonton protocol?
No, I don't. I'm sorry. And, maybe when James talks next, I think, he'll be able to sort of address that question. He might even be able to address it now, but I can't answer that question. No, sorry. I don't know if there is for sure. And maybe James knows that.
Perfect. And of course.
Okay. So sorry. No, but James may be able to address it in his talk. Sorry.
Perfect. So as we wait for any more questions to come in, just to give everyone a heads up, we will be on a break from 11:15 to 11:24. And at 11:25 sharp, we will be starting with the next two talks by Dr. Tim Kieffer and Dr. James Shapiro. Dr. Rajotte, I have a question for you.
Sure.
In this great history that you've shown us of the Institute, what would you say would be the greatest difficulty you faced with regards to where you started and where you are currently?
Yeah, I mean, I think funding, as you heard, funding has always been a little bit of a problem, and we're very fortunate that you know, the Alberta Diabetes Foundation was established, and they funded our trials for 10 years, and then they were the key figures by pledging $10 million that we ended up with the Alberta Diabetes Institute. But funding has always been a little bit of a major problem. The problem also now we have is, you know, how do we support new young scientists? I mean, there's no, right now, there's no ways, if you will, to support scientists that want to do research in diabetes. And it's very difficult to get them, you know, academic appointments. We're fortunate, as I indicated, in 1981, the Alberta Heritage Foundation for Medical Research was established. And their mandate, if you will, is to support young scientists and provide them salary and provide them money to do their research. But that was stopped. And so that has always been a little problem, is how do we continue to support our scientists with hard money position? The other thing we have to do is a lot of our clinical colleagues, we have to give them protected time to do research. And so somehow we have to come up with some dollars to give them protected time. But it sounds like it's money, and it is money in large part, you know, to move the field forward.
I did hear a bit of a concerning stat that with regards to percent of GDP invested into research, Canada is not even in the top 20.
That's right.
Do you have any suggestions with regards to that? What specifically would need to change to bring about better policies for supporting research?
Well, I mean, as you know, and you've heard, I mean, even at the province here, I mean, you know, there's the University of Alberta is going through major changes. And in fact, I think we have to come up with $100 million cut in our budget. So that's going to be a major problem. So I think, you know, both the federal government and the provincial government has to look carefully and say, you know, research is the engine that drives the economy. I mean, when you get a research grant, it not only pays for, you know, the many people, but it helps the economy. So somehow we have to convince our politicians that research is important to fund.
Thank you for your insights, Dr. Rajotte. It is 11:15, so we'll be going on our break until 11:24. And please feel free to stay online or rejoin us at 11:25 for Dr. Tiimothy Kieffer, followed by Dr. James Shapiro. Thank you.
Welcome back, everyone. We are continuing now with the ADI event in celebration of the discovery of insulin. And I'm Stephanie. I'm a graduate student and also a member of the ADI trainee working group. It is my pleasure to introduce now the next two speakers for today's event. And just as a reminder, you can post your questions at any time in the Q&A box. So our next speaker is Dr. Tiimothy Kieffer. Dr. Kieffer did his PhD in physiology at the University of British Columbia, where he's currently a professor and leader of the Diabetes Research Group. Before this, he held positions as professor and instructor here at the U of A and at the Harvard Medical School. Dr. Kieffer's laboratory is focused on the development of novel gene and cell therapy approaches to treat diabetes. He has co-authored a great amount of peer-reviewed publications and several book chapters and holds many patents. In addition to mentoring numerous students, Dr. Kieffer is an active teacher. He received scholarships and his research has been funded by several agencies, including the Canadian Diabetes Association, the Alberta Heritage Foundation for Medical Research, the Michael Smith Foundation for Health Research, CIHR, Canadian Foundation for Innovation, and the Stem Cell Network. In 2010, he received the Canadian Diabetes Association Young Scientist Award, and in 2018, he was elected as a Fellow of the Canadian Academy of Health Sciences. His talk today is entitled "The Edmonton Protocol Points to Stem Cell for Diabetes." So Dr. Kieffer, the stage is yours.
Great. Thank you very much, Stephanie, for that introduction. And I'd also like to thank Peter Light and the organizers for inviting me to participate in this wonderful half-day event. I really have enjoyed listening to Alison, Krista, Ray, and I'm looking forward to James' talk after mine. So I'm speaking to you from the University of British Columbia, the Life Sciences Center on the Point Grey campus in Vancouver, which sits on the traditional ancestral unceded territory of the Musqueam First Nation. And this is our building right here. So I do have some disclosures relevant to my presentation that I've listed here. And I would like to start out mentioning that I was very fortunate to have my first independent faculty position at the University of Alberta. In part, that's why I've really enjoyed listening to these talks earlier this morning. I had a lab in the Division of Endocrinology and Metabolism, and what you see here is a picture of Anthony Chung, my first postdoc, and our first paper that came out from the University of Alberta. You'll note both Greg Corbett and Ray Rajotte were tremendously helpful. Given that I was setting up a new lab, we really appreciated their help in getting things going. So what did our team do here? Well, Anthony came up with the idea of making surrogate beta cells. We already believed that it would be ideal to get physiological insulin secretion back in patients without having to think about it. And Anthony's idea was to target the gut K cells, which were discovered at UBC, and produce this hormone GIP in a meal-dependent manner. So in humans, when you consume glucose, insulin, of course, is released very quickly in proportion to the amount of glucose, and so is the hormone GIP. So Anthony rationalized that in patients with diabetes, they don't have this insulin response, but still have a robust GIP response coming from those gut K cells. So what if we hijack those cells, engineer them to produce insulin? Could we reestablish automatic meal release of insulin, kind of from making a surrogate beta cell? The approach he used was to create a transgene linking the promoter region of the GIP gene to the insulin gene. And at the University of Alberta, we made transgenic mice with this gene and found it worked as described. Again, Greg and Ray helped us to demonstrate in their histology core that we now had mice with human insulin in their gut, specifically localized with GIP in the K cells. So then what Anthony did is gave these mice STZ. This is a toxin that kills the beta cells and normally renders them diabetic, but we were delighted to see that these mice with insulin coming from their gut were not diabetic and could clear a glucose challenge just like healthy controls. So that was kind of our foray in the surrogate beta cell or insulin replacement strategy field. My lab then moved to the University of British Columbia, and we continued that work. Here's Majid Mojibian, who wanted to study this in the setting of autoimmune diabetes. So if we have insulin coming from the gut in this mouse model called the NOD mouse, if the immune cells are killing the insulin-producing beta cells, well, they also go after these cells in the gut. We acknowledge Peter Dickey, who was tremendous at the University of Alberta for making these novel mice for us on the NOD background. And what we found is that, again, the animals were protected from diabetes. And interestingly, the insulin in the gut almost seemed to tolerize the animals to insulin. So there was less attack on the pancreatic beta cells. So we think perhaps targeting the gut for insulin production not only could be considered a therapy, but maybe even offer protection to prevent diabetes onset. Majid really wanted to test this further, and he created animals that are a knockout for insulin, so they cannot make any insulin in their beta cells. This is the mouse here that has insulin coming from its K cells, and essentially lives its whole life without insulin coming from its pancreas, lives off the insulin coming from the gut K cells, and you can see it's healthy and not experiencing diabetes. So we really hope to move this forward to clinical trials. Anthony Chung, that postdoc who led this work in my lab, started the company enGene, which is now located in Montreal. And the hope someday is that we can deliver that gene to the gut K cells using a gene pill, if you will. So that's ongoing work. So we published that first study in 2000 from the University of Alberta. At the same time, the paper came out that you've heard a lot about from Ray earlier, New England Journal of Medicine 2000. I was really fortunate to share a lab in endocrinology with Eddie Ryan. Eddie was the endocrinologist following the patients, and I remember vividly his excitement when he described to me how these patients had this remarkable glucose control after islet transplant, and seeing the results firsthand for myself really solidified in my mind that this could be the long sought after cure for diabetes. You've seen Ray explain how this procedure evolved and how it works. I think it's interesting, we call it a transplant, but it's really an infusion of this highly purified, just a few milliliters or teaspoons of cells. And perhaps he didn't show the remarkable effects on revealed by continuous glucose monitoring. So here's a patient on insulin. You can see hyperglycemia and the worrisome hypoglycemia beyond the level of detection of this meter, the target area being in gray, and you can see what happens after islet transplant. It really is remarkable. And I think this work coming out of Edmonton really was seminal in motivating a lot of us around the world to start working towards coming up with a better supply of cells, and also dealing with that necessity of chronic immunosuppression that comes when you're taking cells from a donor. So at that time, I really became interested in stem cells being a potential source of insulin-producing cells. We know pluripotent stem cells can replicate, so there's an unlimited supply. And we know that they can turn into any different cell type in our body. And what really fascinated me was seeing things like this. So this is human pluripotent stem cells that have spontaneously differentiated. And you can see when I play this, they've turned into beating heart cells. So pluripotent stem cells growing in a dish and just decided they wanted to become beating heart cells. And of course, now researchers have figured out how to control this, and we can buy medias and do this in a very controlled fashion in the lab. But this got me really excited to think, okay, we don't want to make beating heart cells. Can we make insulin producing cells? So the concept here is, instead of having one or two organ donors and purifying those islets to treat one patient, if we start with stem cells, which we can cryopreserve quite easily, we could then thaw a vial, expand it, differentiate those, cryopreserve them again perhaps, and easily treat potentially thousands of patients. In fact, with one vial of stem cells, in theory, if we expanded it enough, we could treat everybody on the planet with diabetes if we could figure out how to do this process. And so the approach we're pursuing involves kind of replicating the stages of human pancreatic development in the lab using these human pluripotent stem cells. So we add, so this is showing the normal process of pancreas development. It takes at least 9 months. Even when we're born, our beta cells aren't mature. But we're trying to replicate this in the lab by taking these pluripotent stem cells and selectively adding different small molecules and growth factors to the cells to coax them specifically down the pancreatic lineage. What's interesting is that we seem to be finding that we only need to go to stage 4, this pancreatic endoderm or pancreatic progenitor cells, and these can then be implanted, so at stage 4, into a subject, and then they will continue their development and maturation for several weeks or months post-implant. So much of my talk will talk about work we're doing with these stage 4 pancreatic progenitor cells, but then I'll also talk about the work we're doing to try to mature the cells further in the dish before we implant them. So you'll notice 2 key transcription factors. These are key markers, PDX1 and NKX6.1, and these are markers we focused on to try to make these pancreatic progenitors. And we did that because if we look at human fetal pancreas, as shown here on the top, the cells that will go on to form the pancreatic islets really have abundant expression of those 2 key transcription factors in the nucleus. So with human stem cells in culture, in about 2 weeks following this particular recipe, we can make aggregates of cells that resemble human fetal pancreas at around 9 weeks of gestation. At that time, the human fetal pancreas does make some insulin, shown here in red, and also glucagon in green, but not very much compared to the mature adult pancreas. And a lot of the hormones are co-localized, so they're immature cells. And you can see the stem cell generated material looks kind of similar in that regard. So what happens if we implant this material in mice? Here I'm showing you graphs of cells put in the kidney capsule. Ray talked about that as being one site that's used in animal models. So first on the right is adult human pancreas for a control. Most of it, of course, is amylase positive in red because it's the exocrine pancreas. You can see the islets are marked in blue by the panendocrine marker chromogranin, and CK19 marks the ductal cells. So in contrast to adult human pancreas, you can see we have largely the endocrine islet shown in blue and ductal cells shown in green. So what physiological effects does that have on animal models? So I'll show you some work done by Jenny Bruin in the lab. She's now an independent PI at Carleton University, but really did a lot of the work that I'll share with you today in my lab. She was outstanding, highly productive postdoctoral fellow. So she, in this study, compared implant of these pancreatic progenitors in mice versus rats using relatively similar doses of cells. Despite using similar doses of cells, we found, interestingly, similar levels of human C-peptide. I point that out because remember the rats are about 10 times larger than the mice and they received a similar dose of cells. So that was interesting. The rats' values for human C-peptide are shown on the top in purple and blood glucose below in purple. You can see the mouse values are highlighted in green. The rats have more human-like blood sugar levels, very good glucose tolerance. And you'll note that earlier in the rats, we detected the maturation of these pancreatic progenitors producing meal-regulated human C-peptide. So same cells, same day going in mice and rats seem to be giving different rates of maturation. Moreover, when we looked at the graphs, we noticed that in the kidney graft, the rat recipients of these cells had a bias towards the insulin-producing beta cells shown in red, 3 different rats here, compared to the mouse, where there was a lower ratio of insulin relative to glucagon shown in green. So same species or same cells, same day, yet different outcomes depending upon the species. Jenny also wanted to look at the effects of thyroid hormones, given that many patients with diabetes have abnormal thyroid function, and she designed this study to examine the effects of periods of hypothyroid, either chronic or acute, or hyperthyroid. And the results were quite telling. We found that both acute or chronic hypothyroid impaired the maturation of the implanted cells, as evident from the dramatic reduction in the output of human C-peptide from these cells in the mice. Moreover, when we took the grafts out, in this case, they were in macroencapsulation devices that I will introduce you to shortly. They had similar amounts of endocrine cells marked in red by synaptophysin, but you can see in the euthyroid group, there was clearly more insulin than in the hypothyroid. So having low thyroid hormone certainly seems to impair the maturation of these pancreatic progenitors. Nellie Saber, who worked on a lot of these studies with Jenny Bruin, also led her own study asking the question, what impact would pregnancy have on these pancreatic progenitors? So she compared the maturation in pregnant or non-pregnant females and included males as a control. And what was really surprising to us is while we noticed no impact of the pregnancy, we found that the cells put into females matured much faster than they did in the males. Again, same cell, same day, very different outcomes. So I'll just focus your attention here. Human C-peptide levels in the males. At the same time, here's the females, whether there's a pregnancy or not. You can see they have higher C-peptide levels and it's meal responsive already at 13 weeks. I mean, you can see even a more dramatic shift in human C-peptide output and meal responsiveness when we looked at 21 weeks. So certainly sex seems to affect in the mouse model the maturation of these cells. So for those reasons, we've continued to endeavor to produce more mature cells in the lab with the thought that perhaps that would allow us to implant cells that would be less susceptible to environmental differences that we may see clinically. And again, Jenny Bruin has been instrumental in this work in my lab. And also this work is largely the result of the efforts by Ali Razania to come up with protocols during his time working at Betalogics, subsequently at ViaCyte, and now he's leading a group at CRISPR Therapeutics and really is, I believe, the mastermind of this field. And I've been really fortunate to work with him. So here you can see this protocol. It certainly takes longer to make stage 6 or stage 7 cells compared to pancreatic progenitors, but we're rewarded by doing that by virtue of the fact that the cells now more resemble human islets. So now they have insulin in cells that don't produce glucagon. They have key transcription factors, for example, MOF-A. Even at high resolution EM, we can see the cells have insulin stored in secretory granules now, like mature adult human beta cells. And we can show glucose-induced insulin secretion from these cells before implant. I'll just demonstrate one potential advantage here in a head-to-head study of implanting stage 6 cells compared to stage 4 cells. So if you look down here, this is the result of implanting mice that have diabetes with 5 million stage 4 cells compared to just 2 million stage 6 cells. And you can see the stage 6 cells work quicker and clearly more robust, improve glucose tolerance more, and that's due to the greater output of insulin coming from these more mature cells. And if we continue to stage 7, they even work quicker. Now in a few weeks, we get very robust human C-peptide, and we know it's coming from our cells, not only due to the specificity of the assay, but if we remove the graft-bearing kidney, the animals become diabetic again, as shown here, and there's a loss of that human C-peptide in the secretion. So now we are further refining the protocol. This is Priya Iwarima in the lab. You heard Krista Lamb talk briefly about her in the presentation. She's a PhD student in the lab. She's now able to make stage 6 cells that respond very nicely to glucose plus incretin hormone in this parafusion assay. You heard Ray talk about dithazone staining. It's not only useful for marking the human islets and distinguishing them from the exocrine, which appears white, but it's also useful for us to show that when we take the cells from the bioreactors at different stages, unlike the stage 5 cells, which don't have that crystallized insulin with the zinc for the dithazone to bind, by stage 6 and stage 7, we get robust insulin-producing cells, and you can see we can make highly purified cells even more pure than what we can get from the human islet preparations. So aside from scale up, one of the things we need to deal with is the immunity, both the response from the immune cells to that foreign tissue, but also, of course, if we're targeting type 1 diabetes, it's an autoimmune disease. And aside from the approach I mentioned with targeting the gut K cells, we think if we put our stem cell-derived cells into patients, they will undergo a rapid demise if we don't protect them from these fire-breathing dragons or potent immune cells. I'll tell you a few approaches we're doing. So we're collaborating with a Vancouver company called Aspect Biosystems to explore the potential of incorporating our differentiated stem cells into an immunoprotective implantable device that's generated with the Aspect Biosystems innovative 3D bioprinter. So with that technology, this is a printhead shown here, we can print multiple different biomaterials loaded with therapeutic cells in real time to create fibers, which are then the building blocks of implantable devices. So these dispensed cell-loaded fibers are directly patterned. An example shown here into a 3D macro scale therapeutic device, and using their software, we can design the structure and composition of these to meet our specific requirements. So using an immune protective outer shell layer that's shown here in blue, we are working to enable the long-term survival of the cells without the need for immunosuppression. So the blue has biomaterials with selective permeability, as noted here. Our cells would be in this core shown in pink. And we can, again, customize this for the dose, the implant site, and considerations for the shape of the design, considering things like vascularization, which are critical for cell survival. So here you can see an example of a 3D fiber that's been printed with alginate. I actually dabbled a little bit with alginate while I worked in Edmonton and learned about it from Rajotte and Greg Corbett. We've tested the survival and function of these islets by implanting the fibers in the intraperitoneal cavity of mice. 31 days later, post-implant, the cells are viable and healthy, marked by this viability stain in green. And again, you can see they look quite good with their dithazone stain, again, following 31 days of being in the intraperitoneal cavity of the mice. And when we retrieve them, they also show good glucose-induced insulin secretion. In terms of diabetes, we again use the STZ model to render the animals diabetic, implant a single fiber containing the human islets, in this case, in the mice. And you can see diabetes was rapidly reversed by the implanted cells. And that's commensurate with an increase from undetectable levels to a few nanogram per mil human C-peptide. The animals also have superior glucose control even compared to healthy control animals. So we know these human islets are functioning in these devices. So we also see excellent function when we take them out if we do a glucose-induced insulin secretion. Interestingly, the stimulation index increases, so they're very happy in these devices, and we see good glucose responses that are more consistent to the printed devices prior to implant, and very low basal secretion which is a very good sign of healthy islets, as stressed islets tend to have increased basal secretion even at low glucose. We've also made similar fibers from stem cells derived from islets, stem cell derived islets as shown here. You can see the dithazone stain. After 7 days in culture post-printing, we get excellent glucose-induced insulin secretion, indicating that our stem cell derived islets are functional post-printing. When we implant them, we're seeing good, robust increasing human C-peptide in the animals. When we take one out, there's very good viability here post-explant. So these studies are encouraging us to continue to pursue this, and we're working closely with Aspect to do preclinical studies, and hopefully in the future we can get into some patients to see if we get similar results with this approach. We're also continuing to explore the use of macroencapsulation devices. This is a Therasite device here, developed by Baxter in the 80s. It's like a tea bag. You load your cells in. Oxygen and nutrients can get through this inner membrane, outer membrane, but this inner membrane is cell impermeable. So the cells are in here. You've got these 3 different membranes, and oxygen and nutrients can get through these membranes. You can see they get highly vascularized after implant, but the immune cells will not be able to get through this inner barrier. At least that's the idea. Again, Jenny Bruin has done a lot of this work in my lab. This is a device cross-section at low mag. You can see high magnification of the region in the dotted line shown here, an H&E stain. These were pancreatic progenitors. So islets don't survive very well, but our pancreatic progenitors seem very robust. And I should mention Andrew Pepper has done some really fantastic work doing similar studies at the U of A with James Shapiro. So we have endocrine cells surviving. The pancreatic progenitors mature. So here's pre-implant, post-implant. We can see islet-like cells in the device. And I just want to show another cross-section of one of these devices here, stained for insulin, glucagon, and somatostatin. Human pancreas on the right, stained under the same conditions. And you can see the remarkable resemblance. It's like these devices which are designed to go under the skin contain a sheet of pancreatic islet material. So if we take a small device about the size of your fingernail, implant that in diabetic animals, you can see that we can quickly lower blood sugar in the animals. I say quick, it takes about 3 to 4 months. And as blood sugar is coming down, there's an increase in human C-peptide. So these animals do not require insulin therapy. They live the rest of their lives with these devices, taking over insulin production from the beta cells that we've eliminated, in this case by low dose STZ. Kara Ellis, you may recognize, she trained with Greg Corbett. We were really fortunate to have her join our lab as a postdoctoral fellow here at UBC. And she's continued kind of where Jenny Bruin left off, looking at macroencapsulation devices. And here she's comparing devices that are either intact or devices that have portals which allow direct vascularization of the cells within. She compared those both in mice and rats using the doses of cells shown here. So first, looking at the mice, you can see in these healthy mice, interestingly, after a few months, there's a lowering of blood sugar, and we now appreciate that it's lowering it to a human set point. These are human cells, and normally they wouldn't be continuously exposed to glucose this high. So once they start functioning, they take over control of blood sugar, and both devices clearly work in the mice and lower it to around, say, 5 millimolar on average. The rats are already hovering around 5, maybe 6 millimolar, but you can see a little bit of lowering only in the animals that had the directly vascularized graft in the rats. We looked at glucose tolerance at different weeks post-implant, and you can see as the cells matured in these healthy animals, they actually became better than normal in terms of their glucose tolerance. So in the mice, both recipients of the pancreatic progenitors from the stem cells in immunoprotective or directly vascularized had kind of super glucose control, better than the controls. In contrast, the rats already have excellent glucose control, more similar to humans. And you can see the directly vascularized, for the most part, was able to slightly improve upon that. And that reflects the human C-peptide in the rats. In the directly vascularized, you can see, with the exception of one that worked moderately, the animals, for the rats that received the intact immunoprotective device, there was really very little function, whereas the directly vascularized with those portals clearly was able to work, and both devices worked in the mice. We actually think now that the rats could be a better predictor of what would happen in humans compared to the mice. So we're involving them more in our studies. Kara did this high resolution glucose challenge, which showed something else interesting for us. Again, in the mice where both devices worked, we saw better glucose control in the directly vascularized. And when she did this high resolution analysis, she found that there was much more rapid and robust human C-peptide production from the cells when they were in the directly vascularized type of device. So I think these are important data to inform future designs. As I said, in the mice, the human cells take over. These were non-diabetic animals. And if we look at mouse C-peptide now, we see that it's essentially shut off in the recipients of these human stem cells. So there's no need for the mouse beta cells to work anymore because of this lower glucose achieved with the human cells. Similarly with the rats, and a more modest reduction in the immunoprotective device, which didn't work as well. Now, these devices were developed by the California company called ViaCyte. I'm sure many of you are familiar with it. I've just been describing data with the PEC and CAP and the PEC Direct type of device, which has these portals. This, of course, requires the use of immunosuppression because now we've penetrated that barrier to immune cells. But ViaCyte is working with CRISPR Therapeutics to use genetically cloaked cells with this device in the hopes that they can move towards a therapy without the use of immunosuppression, which I think for all of us in the field is our long-term goal. So next, I'll share with you the results of an ongoing clinical trial testing the safety, tolerability, and efficacy with ViaCyte's combination product of pancreatic progenitors and these macroencapsulation devices with the portals. And this work is largely being led by these two. So this is David Thompson. He's the endocrinologist who's been recruiting and carefully following these patients post-implant. Adam Ramsey has just completed his MD PhD at UBC and really has been instrumental in collecting and analyzing a lot of the data and samples from these patients. So I will present quickly, keeping an eye on the time here, results from 15 patients. Again, they had immunosuppression because of the portals, very similar immunosuppression to Islet transplant recipients. They received 2 to 4 of these devices that look like this, as well as some smaller sentinel devices that can be taken out at different times to get a look at what's happening within the devices. And they are put, again, subcutaneously in the forearm or flank. And I'm sure Dr. Shapiro will be telling us more about this procedure, as Edmonton is also participating in this trial. So quickly through these 4 panels. So in all of the panels, individual patients are shown by the different colored lines. Again, there's 15 patients. The multiple level regression is shown by this bolded black line, and the standard error of the mean is shaded in gray. So first, body weight of individual patients tracked relative to baseline, and you can see it was stable. Hypoglycemic awareness over the study was assessed using the Clark hypoglycemia score, and patients had improved awareness over the duration of the follow-up. Hemoglobin A1C was monitored and was stable over time. Patient insulin requirements were logged from prior to implant and throughout the study, and you can see there was reduced insulin requirements looking at the slope of this line post-implant. Patient blood glucose was tracked using continuous glucose monitoring, and here I'm showing you the time and range of hypoglycemia, less than 3.9, euglycemia, target range 3.9 to 10 millimolar, and hyperglycemia greater than 10. And you can see that there was significantly more time in the target range here, 3.9 to 10 millimolar, and significantly less time in the hyperglycemic range as shown on the right panel. So I think for us, Adam, David, and I got most excited about these data. So these are individual patients' serum C-peptide levels over 4 hours in response to a mixed meal, either pre-implant, the circles, 26 weeks post-implant, the squares, or 52 weeks post-implant, the triangles. And you can see for most of the patients, we got a progressive increase in human C-peptide secretion, albeit at variable levels. You can see the y-axis scales are different. I should mention that in 2 of the patients, they completed mixed meal tolerance tests after graft removal, and in both of them, we no longer could detect C-peptide, or it was at their pre-levels, convincing us that what we're seeing here must be coming from these pancreatic progenitor cells that have presumably matured into functional insulin-producing cells. Indeed, when we take out a graft, we can see pockets of surviving cells in the device. So here, synaptophysin marks endocrine cells in green, and we've got CK19 marking the ductal cells in red. So you can see compared to pre-transplant, now we're left with these discrete pockets of cells, at least in the areas that we have assessed thus far. Unlike the unimplanted device, where we didn't really see, as expected, a good amount of insulin and glucagon, single positive cells, in these surviving pockets of cells, we could clearly see insulin immunoreactive cells that didn't have glucagon, and vice versa, glucagon immunoreactive cells that didn't produce insulin. So those pancreatic progenitors have not only survived, they've continued to differentiate and mature, and that's also exemplified here when we look for key transcription factors in the nucleus of the cells, and you can see them here, NKX 2.2, NKX 6.1, PDX1. It's now restricted for the most part to the insulin-producing cells, with the exception of PDX1, and that's similar to human islets, and most notably, perhaps, pre-implant, there's no IAPP in these cells, but after they mature, there is, similar to human pancreas. And MOF-A is absent pre-transplant, but we gain it in the insulin immunoreactive cells post-implant, again, suggesting that the cells have matured. So to summarize what we've seen in this clinical trial with ViaCyte over 1 year, we have improved glucose control, we have reduced insulin requirements. No, these patients aren't on insulin, off insulin, sorry, and that simply reflects that although we're seeing this increase in fasting C-peptide and meal responsive C-peptide, clearly we need to increase it more before we're going to see patients completely off insulin. Nevertheless, I'm excited by these findings because we've got surviving insulin immunoreactive cells that had a mature beta cell phenotype, including nuclear MOF-A. And so we feel these data provide evidence that stem cell-derived pancreatic progenitors can mature into glucose-responsive, insulin-producing mature beta cells in patients with type 1 diabetes. So just to close, I think, again, coming from those days in 1999 and 2000 when Ray Rajotte's team, working with James Shapiro, taught us that the clinical path for cell therapy is now proven with islet transplant. There's tremendous promise, I think, of using stem cells now. But what is the optimal cell preparation? I've talked a lot about pancreatic progenitors, but I also talked about the feasibility now of working with cells that have matured further prior to implant, but it remains to be determined how they will function. Remains to be determined where we should implant those cells. And I'm sure Dr. Shapiro will talk more about that. How should we protect those cells from immune attack? Again, ultimately, we don't want to be using chronic immunosuppression. I think there's a lot of merit in thinking about encapsulation, whether it's micro-encapsulation, macro-encapsulation, or perhaps making a customized 3D cell-containing patch using Aspect Biosystems technology. And finally, I think it's a really exciting time. We've just seen the Nobel Prize for genome engineering with CRISPR, and I think we're only limited by our imagination now to think about, can we genome engineer the cells to protect them from immune cells, to enhance their survival, and perhaps also to consider their safety. And I'm just going to skip now to the acknowledgements and really thank the group at Vancouver that's been outstanding to work with on the clinical studies, which will continue. And I predict there's going to be a lot of ongoing clinical activity in the future. And we've got a great team here in Vancouver to lead that work. These are the lab members who have participated in these ongoing studies we're doing with differentiating stem cells. We've been really fortunate to collaborate with Betalogics, Janssen, and ViaCyte and Aspect Biosystems, fantastic collaborators here at UBC. And finally, I would like to finish by acknowledging the funding that really started from my time in Edmonton and transferred here to Vancouver that made this work possible. So thanks very much for your attention, and hopefully I've left time for some questions. If not, I'll answer them in the chat.
Thank you, Dr. Kieffer, for this exciting presentation. So we do have time for one or two questions. And the first question comes from Cheryl. And first, she thanks you for the presentation and says the technology that you are working with is really amazing. And she wants to know if you know of any research regarding hormone levels and insulin requirements. She explains that during her life, she associates diagnosis of diabetes with puberty and difficulties in blood sugar control with menopause.
Yeah, so that's a great question, Cheryl. And, you know, it's been interacting with individuals with type 1 diabetes, and even having people in the lab who have type 1 diabetes, that I think we've really become aware of how challenging it is to manage glucose levels. It's much more than simply looking at, you know, the meals that are consumed and trying to predict how much insulin requirement. As Cheryl has articulated, various hormones can impact insulin requirements, exercise, diet. There's all kinds of things that certainly there's a lot of research looking into this. And that's why we feel that ultimately, if we can recreate the remarkable pancreatic islet function in patients, the vision of Ray Rajotte and others, that that's going to do the best for the patient, because we assume that those cells should be able to respond to those different requirements that we face living our lives, and hopefully can restore normal glucose homeostasis. So thank you, Cheryl, for that question.
Okay, so in the matter of time, we will move to the next presentation. But if you can, please, Dr. Kieffer, there are some other questions in the chat box.
Certainly. Thanks, Stephanie.
Thank you. So our next speaker is Dr. James Shapiro. And Dr. Shapiro was born in Leeds, England. He's the son of a family doctor, and he developed a long-standing interest in islet cell transplantation as a medical student. He has been on faculty at the U of A since 1998, where he now holds the Canada Research Chair in Transplantation Surgery and Regenerative Medicine, and works as a busy liver transplant and cancer surgeon. He was the lead investigator on the famous Edmonton Protocol, cell transplant treatment for diabetes, and was the first in Canada to start clinical trials with human stem cell-derived insulin-secreting cells. His many awards include the Hunterian Medal for the Royal College of Surgeons of England, a gold medal from the Governor General and from the Royal College of Physicians and Surgeons of Canada, and a Lifetime Achievement Award from Diabetes Canada. He's a Fellow of the Royal Society of Canada and a Fellow of the Canadian Academy of Health Sciences. His talk for today is entitled Cell Transplantation from the Beginning to the End of Insulin.
Thank you, Stephanie, and thank you to Peter Light and to the committee for putting together a wonderful celebration of 100 years of the discovery of insulin. My job this morning, I think, is fairly easy in being able to sum up some really wonderful talks reviewing the history that Alison Lee provided, the journey that Tim just took us on, and Ray and Krista. So I think my job is easy, and forgive me if there's a significant overlap between what you just heard and the journey ahead to the future. Well, I've been very fortunate to be here at the University of Alberta since 1993, where I came to learn liver transplantation and also work with Noam Knieteman and Ray Rajotte in islet cell transplantation. And I'm going to take you on a summary journey this morning on cell transplantation from the beginning to the end of insulin. So, as you heard from Alison earlier, the treatments for diabetes before the discovery of insulin were fairly horrific. The eating of pancreatic digest was carried out by some, but this Allen diet was about the only treatment available for patients with type 1 diabetes that did allow some extension of life span for a very short period of time. But before the 1920s, this disease, diabetes, was regarded as with the same horror that we deal with late-stage pancreatic cancer today. So these medical interventions brought a few extra years, but always led to a slow and excruciating death, which was inevitable. And this liquefaction of flesh and bones into urine, well described by many manuscripts, and even the BC era, really was revolutionized by the discovery of insulin. And here's a picture of Banting with his business card in 1920 in the summer there, where he was only able to earn $4 a month as a family physician. He didn't get a job as a surgeon. So he was teaching physiology at the Western University. And this was his big idea in October, Sunday evening, 2 a.m., 1920, when he was too tired to spell diabetes correctly. And in fact, several of his notebooks for several months afterwards continued to spell it the same way. But he had this idea of ligating the pancreatic duct in dogs. And as you've heard from Alison, that eventually that idea didn't really pan out, but at the time it really led to the opportunity of being able to extract insulin. And this was the paper that he read the night before that lecture that describes stones in the pancreatic duct that led to the possibility of being able to separate the islets out from the rest of the gland. So these are some of Banting's notebooks. This is from June the 14th, 1921, so 100 years old. You can see here his description and discussions with MacLeod, and with MacLeod's parting instructions to give the dogs the diabetes extracts and either deliver it intraperitoneally or put it under the skin subcutaneously, either preparing the whole remnant of the graft subcutaneously, and all their ideas that they were working on at the time. But one of the ideas was to prepare emulsions of the whole remnant of the pancreas in one shot, which is essentially what we've done for the last 20 years with islet cell transplantation in a very crude way. So 100 years ago today, Banting's notebook, June the 16th, Thursday, they were still working on turtles using the Lewis Benedict method to measure blood glucose in 0.2 mils of blood in these turtles. And these are some of the records from that time. And these are very, very intriguing to look at. And the fact that they were able to get these to work eventually with the pancreatic extract in dogs, as you've heard from Alison, was a miracle that led to the first patient, Joe Gilchrist, who was a Banting's classmate at medical school, who was diagnosed with diabetes in early 1917, receiving the first dose of this insulin extract. That was on December 20th. It was given by mouth, as you can see here. The idea then was that perhaps this pancreatic extract could be given by mouth, absorbed in the intestinal tract, and lead to clinical benefit and reversal of diabetes. But clearly, from Banting's notes here, there was no benefit at all. And the first patient that you heard from Alison also, Leonard Thompson, came into hospital, 14-year-old, 29.5 kilograms, all skin and bones, described by the house officer there. And on January the 11th, 1922, received this 15 cc's of what was called McLeod serum at that time, thick brown muck, injected into the buttock. The blood sugar did fall slightly, but there was no clinical benefit. And there was a complication of the very first treatment of insulin. So a sterile abscess developed on day 7. If that had stopped the process, we wouldn't have insulin today. And then you've heard about Bert Collett, coming to Toronto with his Rockefeller Scholarship and really being the key that unlocked the purification of insulin. And he also described the unit of insulin as being the amount required to induce fatal hypoglycemia in rabbits. This was the first paper published in the Canadian Medical Association Journal in 1922, describing Banting, Best, and Collett's work. And the first patient, Leonard Thompson, now deriving clinical benefit with a fall in blood sugar from 29 to 6.7 millimoles per liter on continued treatment for 10 days. And that really opened up the frontiers of a treatment for diabetes that has been with us for the past 100 years and has really led to revolutionary outcomes and clearly the longer-term survival for patients with this disease. At the time, it was thought this might be a cure for diabetes, but clearly it wasn't. Banting very clearly stated that in his Nobel laureate lecture. The sale of the patent to the University of Toronto for $1 is well documented. The concept of being able to mass manufacture this insulin over the next few years with the Connaught laboratories clearly allowed patients to be treated. And today we celebrate this with a stamp 100 years later, celebrating the Canadian discovery of insulin. So I take this from Professor Sir John Bell's lecture that he gave University of Alberta a few weeks ago, where he describes all the advances in medicine that have occurred in the last 30 years. The cardiovascular mortality is reduced by 70%. There are now, as of today, 2.26 billion doses of a COVID vaccine administered just 1.5 years into a lethal pandemic of that disease. There are 3 chronic infections that are controlled and cleared, being HIV, hepatitis B, and hepatitis C. And the University of Alberta's Lorne Tyrrell and Michael Horton played very key roles in both of those discoveries that led to treatments and cures for those diseases. We have 7 new drugs that are active for multiple sclerosis, a panoply of drugs that are very effective now in rheumatoid arthritis as anti-inflammatories. The human genome has been sequenced over 200,000 times, and we have a cure for cystic fibrosis. We have very effective cancer immunotherapies, such as those for what was, when I was a medical student, a lethal disease, melanoma, can now be treated in many patients and potentially cured. And overall lifespan, life expectancy has increased by 10%. So there's been a lot of progress when you pause for a moment and see what's happened over the last 30 years. We've got remarkable advances in the care of patients with diabetes, new insulins, better human insulins, different absorption profiles, continuous glucose monitoring devices, closed loop pumps, and a whole panoply also of new drugs that are effective in the treatment of type 2 diabetes. But what I'm most excited about are the new therapeutics available, cell replacement therapy, as of the Edmonton Protocol and beyond, and the regenerative medicine approaches that Dr. Kieffer describes so eloquently, has made so many brilliant contributions to over the recent years, and has allowed this, I think, to be a real possibility for our patients. So we know that insulin prevents death, but it is not a cure for diabetes. And every 6 seconds, somebody dies of this condition. There are estimated 463 million people across the world today that have this condition, and this is going to grow massively to 700 million or beyond by 2045. It's the most expensive of all the diseases that our governments have to deal with. And globally, we spend $1.3 trillion in managing diabetes and its complications every year. So this is a disease that is hungry for a cure. Advances in wearable therapeutics for diabetes, like these closed-loop insulin and glucose systems, are advancing. But you can see that despite the variations in the blood glucose level that are improved from baseline in patients, these are still far, far from normal. And patients continue to have fluctuations in glucose, even with the latest bionic pancreas technologies. And many patients don't enjoy wearing these technologies. They can limit their lifestyle. So why is injected insulin limited in its efficacy? When we inject insulin, it goes under the skin as a bolus. And this is a CT scan of a patient that's received a bolus of insulin. You can see it sits there. It gets slowly or rapidly absorbed, but it's nothing like the normal physiological response inside the human pancreas. So these are human pancreatic islets that pulsate with insulin release, glucagon release, and somatostatin release every 10 to 15 minutes. And that is a far cry from what we can achieve today with insulin injections of any form. So Tim showed you this picture, but I'm showing it again. The remarkable glycemic control that can be achieved with cellular replacement therapy. Here, a patient receiving an islet transplant has completely normal glycemic control off insulin. So I'll take you on a journey of cell transplantation for diabetes, and it begins at the hospital where I trained to be a surgeon at the Bristol Royal Infirmary in England, southwest England, where a surgeon, Watson Williams, and his assistant Harsant took a 13-year-old boy dying from the ravages of diabetes, and they put a sheep's pancreas in pieces beneath the skin. You can see how desperate they would have been in those days, 27 years before the discovery of insulin. There was nothing else really that could be done, and the idea that you could transplant some cells or some products of the pancreas was a desperate attempt. This was a xenograft, so it had no chance of working, but it clearly was a remarkable experiment. Similar experiments were carried out at where I trained as a medical student at the Royal Victoria Infirmary in Newcastle in England, where a surgeon there, Frederick Charles Pybus, in 1916, shown here in the picture, was taking organ donors essentially from road traffic accident victims. In 1916, he describes in his paper quite well all the details of doing this, transplanting adrenal glands and also grafting pancreas again beneath the skin in patients. And he describes also really elegantly that these cells were maybe worked for a short period of time, then were rejected. They didn't know at that time anything about organ rejection, of course, but he describes the principles of grafting until we understand them better, and I feel we do not understand them at present, especially the chemical factors, then we must continue to fail in such operations. And while we have much more knowledge in this area today, we still face challenges with the immune system and better understanding how to control that. So Ray Rajotte mentioned the wonderful work of Paul E. Lacey and his proof that cell transplantation, islet cell transplantation, could reverse diabetes in rodents. And in this paper in 1973, he showed very clearly that the portal vein was more effective than the subcutaneous or omental sites for implanting islets. And that's really why we ended up with the portal vein as a site for clinical implantation of islet cells, potentially for future stem cells today. And so young Ray Rajotte described it in detail. I won't go through all of his wonderful lecturing work and historical perspective that he brings to this, but his idea to freeze kidneys and then taking that on to freezing islets, and his connection with the St. Louis group and Paul Lacey and many others led to his experiments in Edmonton to freeze and thaw islets for clinical transplantation. And here is a clinical picture of one of the first islet transplants in 1989 with Norm Kneteman, Mark Evans, and Garth Warnock carrying out that transplant, published in The Lancet, describing continued function in that patient. And of course, I read all the papers that Ray Rajotte produced from Edmonton, including the first clinical attempts, and that was one of the reasons that really attracted me to come to Edmonton and also to begin to study this as a medical student. Here I am as a young medical student in Newcastle, working in the evenings for Radio Tyneside to earn a few extra pennies, but working primarily with my mentor, John Farndon, to learn about islet cell transplantation and see if we could alter the immunogenicity of those cells by freezing and thawing using Ray's techniques. So take you on a journey ahead with, I came to Edmonton to learn liver transplantation with Norman Knietemann, but of course rubbed shoulders with Ray, Rajotte, Jonathan Lakey, Greg Corbett, Eddie Ryan, and others. And I was asked to lead the clinical islet cell transplant program after a period of pause. And this was our paper that's been discussed at length. Before the Edmonton protocol, the success rate with islet transplantation at a year was less than 10%. It was fairly dismal. So when I was given this gift of running the islet program, it seemed a bit too edged in some ways. But here we are at 11 o'clock at night, Ray calling the islet digest. And this is in the basement of the SMRI in the islet isolation lab there, which was, of course, Collip's, the same building that Collip worked in after he came back to Edmonton. When we moved forward and carried out a multi-center trial with islet transplantation, the FDA came and visited this laboratory, and Ray showed you pictures of the cockroaches. There really were cockroaches around in the basement. And the FDA said, no, you can't run a GMP multi-center clinical trial from this laboratory. So that's why we did move fairly rapidly from there to the College Plaza. Camillo Ricordi, we had the opportunity of working with Camillo fairly closely when we were having some challenges with islet isolation in Edmonton and the enzymes and his techniques. So Jonathan Lakey and I went down to Miami and spent a sabbatical period with Camillo Ricordi, learning his techniques to see if we could implement those in our planned first-in-human trials. So today, this is some members of the team. You can see on the right, one person that's had more experience than anyone else in the world in isolating human islets now, Tatsuya Kin, with his team, and this process is well established. Ray described it in some detail, so I won't go through it, but basically collagenase enzymes are perfused through the pancreas. There's Doug manually shaking the chamber. We do have modern technology for automating that, but I think the hands of Doug and Tatsuya are probably more effective. And then purifying this on the code, Ray described that very well. Here's Tatsuya and Doug and the team over their 2000th human islet isolation, and we're so fortunate to have these expert individuals that work so well together as a team, doing this day and night, and they've produced the best islets in the world for clinical transplantation. I'll also mention and emphasize a spin-off from this work with Pat McDonald's Islet Core team with James Lyons. There you heard that from Ray. This team has produced fantastic quality human islets for worldwide distribution. And it's also, I think, an amazing accomplishment from the ADI and the funding from Alberta Diabetes Foundation. So the Edmonton protocol now 21 years on, you can see we've carried out over 700 intraportal islet infusions. Interestingly, 2 of our very first 12 patients remain insulin-free, one with his original graft 21 years later. And we wish all patients, we could understand better how to maintain that. Here's some videos of the transplant. It doesn't require surgery. It's carried out by percutaneous access by our interventional radiology experts. And infusing the cells into the liver is very straightforward. It takes around 20 minutes to do it. An important adjunct that we've learned fairly early in the process was to plug the tract with a thrombostatic agent, Avatine paste, so that we can avoid the risk of bleeding afterwards. And it's a very, very straightforward transplant procedure. These are some members of the clinical and research teams that have been involved with the islet cell transplantation team over the years, and the funding opportunities and support that we've received from the early beginnings, where we had to scrimp and scrounge through the hospital and under the radar to get things done. But today we have a very solid team of individuals. Now, I'll emphasize the contributions of Eddie Ryan, who I worked with very closely in selecting our very first patients. Peter Senior took over the role from Eddie in helping us select and monitor our patients afterwards. And Dr. Anna Lamb also joined us with helping to support and monitor and take really great care of the islet transplant patients, along with our surgical colleagues, David Bigham, Khaled Dajani, Blair Anderson, and Norm Knietemann. So we've also had a huge amount of help in moving forward around 20 different clinical research protocols in islet and stem cell transplantation across to the clinic with a trial oversight team. We recently lost Dr. Andrew Malcolm this year. This was a big loss for our team, but he put together a remarkable team of individuals that he worked closely with, and I've had the opportunity to work very closely with since. Faye Akishin, Indri Prawana, Diane Parastu, Reina, and Saira really help to secure the infrastructure that's absolutely needed to move clinical and research trials forward. I'll also emphasize the contributions that our many clinical fellows have made in islet cell and liver transplantation over the years in taking care of our islet cell transplant patients. We've been very fortunate to work with this brilliant team of people, surgeons mainly, that have now are now leaders in their own field in various sites across the world. We looked most recently at our function of our islet cell transplants. 20 years later, you can see that over 60% of patients continue to make durable C-peptide that is sufficient to normalize or near normalize the hemoglobin A1C and prevent risk of dangerous lows. This is a scoring system that we've used in patients that it's called the HYPO score. And patients before transplant have very high risk of hypoglycemia. That's just one of the selection criteria that we used initially. And you can see across 20 years in the red lines compared to the median at the beginning, it's close to zero for nearly 20 years afterwards. So highly significant and meaningful results for patients such as Nina here with her husband, Brent, islet transplant number 252, featured in this month's University Folio magazine. She has type 1 diabetes tattooed on her forearm, and she describes how like many of our patients describe how she hasn't had to take insulin for long periods of time. She's living a life that she could only have dreamed of before her transplant, and every morning she wakes up she feels optimistic, and was so lucky, I think. And this is one of the sides of transplantation that really we take it for granted when we're so close to the microscope or to the details of every day. But transforming lives for patients is not only a side benefit, but it's what we all do as a team. Braulio Marfil-Gaza, who's working with me as a PhD currently in the lab, has analyzed our insulin independence rates in islet and pancreas transplant, and you can see here over 78% of patients can achieve insulin independence for periods of time after an islet cell transplantation, but this is less durable than the red line here for pancreas transplant patients. So most patients with a pancreas can remain insulin-free for many, many years, whereas the islet patients need to return to small amounts of insulin after 5 or 10 years. And that's been a major focus of ours to try and improve that outcome over the course of time. This is showing the control. The hemoglobin A1C is a measure of how well and how tight the blood glucose is over the course of time. And you can see both islet and pancreas transplant patients enjoy very tight glycemic control at levels around or less than 6.5% that really cannot be achieved by any other approach. Even the best insulin pumps and closed-loop systems very rarely get you a hemoglobin A1C less than 7 or 8%, particularly in patients that are at risk with this brittle disease. This is showing how safe the procedure is and the concomitant anti-rejection drugs that are given. The patients can enjoy long survivals despite this, and you can see 20-year survivals even in our patients that are sometimes in our late 60s or 70s when we enroll them in islet transplant can enjoy excellent survival. And if we control that for age here, you can see the islet patients is significantly improved compared to the pancreas patients at 20 years. So this leads us to consider, we've used criteria such as the Clark score, the HYPO score, lability index, the scores that Eddie Ryan developed for us for islet cell transplantation. But these are, like, if I can use the word paternalistic, we tend to select patients based on what we think are objective criteria, but we don't live with this condition. We don't live with diabetes every day. And I think we could learn a lot from our patients and probably allow the patients themselves to be more selective and determine if the risk-benefit is right for that particular patient. We're making those judgment decisions for them. And I think now we can, with this informed long-term data, can allow the patients with full and informed choice to decide if they want to undergo cell transplantation. So we've helped the FDA move forward with allo transplantation. One of our former fellows, Jose Oberholzer, put forward a first FDA donor cell program to get approval from the FDA for islet cell transplantation. We've been very, very fortunate actually to have approval for islet transplantation in Canada and in Edmonton for the past 20 years. And we were talking yesterday with Paul Greenwood, who was chair of the Province-Wide Services Committee in the year 2000, 2001. And he had a very fortuitous breakfast with the health minister at the time. And when the Edmonton Protocol success was announced, they asked what could they do to help that process, and approval, clinical approval for that process very, very early on has helped us immensely in Edmonton. So here's Bob Teskey, a lawyer for the university, throwing away his insulin vials and exchanging them for a small handful of anti-rejection pills every day with a smile on his face. But we know that those anti-rejection pills do have side effects. They increase the risk of certain cancers. They can increase the risk of life-threatening infections, and they can also have side effects on the kidney. So I think what we've showed is proof of concept very clearly that the Edmonton Protocol and islet cell transplantation can potentially be a cure for diabetes. We never pretended, even from the beginning, that this was a cure in itself. But we've shown proof of concept that cell replacement therapy can robustly, in human patients with type 1 diabetes, reverse the diabetic state. And we recognize the two clear challenges ahead of us. The supply of islets and the need for the anti-rejection drugs are limiting factors that prevent us treating children and preventing us moving forward to treat 463 million people across the world with all forms of diabetes today. If we could overcome these two barriers, we could make a lot of progress. We're making some efforts in this regard, and I'll just mention a couple of them. We've been working with Jeff Bluestone that Ray mentioned earlier, through the Immune Tolerance Network and through the generation of regulatory T cells, Tregs, at the University of San Francisco, UCSF site. So we've been sending blood from our islet transplant patients to UCSF, where they manufacture and expand autologous Tregs, and we infuse those back into our patients. And that trial so far, early days, very preliminary, but it's looking to be very promising. We're also working in the lab on trying to use patients as their own GMP facility, walking cheap GMP facilities by using antibodies to expand regulatory T cells. And Braulio Marfolgaza has made a lot of progress with two different antibodies that we've been testing recently, at least in our rodent models, that can also substantially augment the autologous T regulations without having to expand that in the dish. So there's many different approaches moving forward. We've also been working in patients with new onset diabetes and using some of our immune interventions to reverse the autoimmune state and immune reset, essentially. And that is also looking to be promising. We have some patients 2 years out that are still insulin-free with that kind of approach with sustained C-peptide levels. Tim Kieffer really helped set the stage for me for the use of stem cell therapies to replace the cadaveric organ donors for islet cell transplantation. And the initial work from ViaCyte, we've been working with this team for almost 20 years now. Initially, Everett Kroon and Kevin Damour and Ed Beji initially in that company. But today, 20 years later, they really did establish the protocols for development and recapitulation of how the islet develops in a normal human embryo. And what they've done is create a product called PEC-01, which is essentially taken from a single human blastocyst from a discarded IVF clinic embryo. And they did that 20 years ago, and that PEC-01 cell line has been expanded massively, and as Tim mentioned, could potentially deliver a worldwide supply of need for these kind of cells. We carried out our first clinical transplant 8 years ago with ViaCyte using these egg capture technologies, initially without immunosuppression and then with some immunosuppression in non-perforated and perforated devices. We were putting these under the skin in the forearm and in the abdominal wall in our patients. And it proved to be a surprisingly safe technique, even in patients that are immunosuppressed. They tolerated this surgery well. And we were able to demonstrate, I think, fairly good islet function with these transplanted pancreatic progenitors. Important safety data that we've all learned, I think, from these trials is the absence of any off-target teratomas, no cellulitis in our patients, very good graft survival in some cases, but it's not perfect. And I think one of the constraints of these kind of devices, they do facilitate first-in-human trials, but they really also impede the ability to provide massive numbers of cells and grafting like we could potentially if we put these cells into the liver or some other well-vascularized site. Nonetheless, in at least a third of our patients, we can detect measurable C-peptide over the course of time. Tim showed you data similar to this, and it's encouraging. But the question really is, in order to match the amount of function we get with a human islet cell intraportal transplant with these kind of approaches, we would need a massive number of devices. And in some trials now with ViaCyte, we're putting in up to 10 of the therapeutic devices to see if we can come somewhere close to the human islet intraportal engraftment. But I suspect even with that, we're not going to get as close as what we get intraportally. So question is, can we move forward with these kind of cells and move them into the clinic, into the portal vein in a safe site if there's no off-target growth? We've been working with ViaCyte, as Tim mentioned, with Alarizania and others at CRISPR Therapeutics to develop this PET-QT VCTX210 trial. It's moving forward through Health Canada now, and we anticipate that both Edmonton and Vancouver will participate in the first human trials. There was a lot of discussion initially about which kind of what kind of edits would need to be carried out to make these cells compatible without immune system. Should we remove all the alloimmune protein? Should we provide something that would attenuate the host immune response, something that would increase the cell survival, something that would increase vascular ingrowth, and probably importantly, provide some cloaking that would protect against recurrent autoimmunity in patients with type 1 diabetes. So we're very excited. And these are going to be iterative trials, I think, moving forward. And the first 4 edits that are moving forward to the clinic may not be the final versions, but they're certainly moving forward. There's a number of other companies involved with this, and I know noted George Harb. Hi, George. Was on this Zoom today, and his company Vertex has clinical approval for an FDA trial for intraportal delivery of an embryonic stem cell product, VX880, in patients with type 1 diabetes together with immunosuppression. And that really demonstrates that the FDA is allowing the intraportal delivery of these differentiated allogeneic embryonic stem cell derived islets to be carried out for the first time. We're very excited to see the outcomes of those trials, as well as other trials such as the company Ceraxis with their SR1423 cell line that will be implanted subcutaneously in macroencapsulation devices. Well, Tim Kieffer introduced this, and Tim, I got to give you absolute credit for getting me interested in this area of personalized iPS-derived islet therapy. You spent a year with Shinya Yamanaka in Kyoto in Japan, learning the techniques for iPS stem cell generation, and I've been really excited to have the opportunity and continue to work with you and learn from you as we try to take this kind of approach forward to the clinic. A few years ago, I thought this was all going to be alchemy and nonsense. We'd never be able to achieve personalized medicine type therapies for patients. We recognize there are many, many challenges ahead. Not only the cost of some of these therapies is around $1 million per preparation today, but we anticipate with some significant advances in the science that these costs will be massively reduced to around $20,000 or $30,000 perhaps per patient preparation in the near future. The possibility that iPS cells could be unlocked was first described by, and the Nobel Prize went to John Gordon and Shinya Yamanaka for these marvelous, incredible discoveries for pluripotent stem cells. And interestingly, this is from the Eton College where John Gordon was last in his class and marked 223 out of 500. Was told that he would never make it in science, and he should definitely not consider that quite ridiculous. And so don't be discouraged if your marks are low and you don't necessarily fit into the community. So Shinya Yamanaka's Yamanaka factors allow adult human cells to be unlocked and turned back to a more malleable state. And Tim also showed you beating heart cells derived here from skin cells from Yamanaka's original publication. But using the combination of the Yamanaka factors to generate iPS with the techniques that ViaCyte developed for the differentiation of stem cells into islet cells and the more mature process that Tim has really described to you so elegantly and has contributed so greatly in the science and understanding and process of this, we're really excited to be able to think about taking this forward now to clinic. Many of these protocols have been well described. Dieter Igley and others have really created recipe books for how to do this in the lab. We've been able to copy these kind of processes, and here are the contributors. Tim here at Christina Nostro, you heard of previously in Toronto, Matthias Hebrock, Doug Melton, and others that have contributed in so many ways to the advances. So I got so excited about this, I converted my own lab over to being a stem cell lab a year ago, and I was very lucky to be able to bring in Dr. Nadish Dadic to lead the development of the IPS generation in islets, and he's been able to replicate, I think, very successfully so far these kind of protocols in the lab, and we've got a very nice lab now thanks to the ADI to be able to move this work forward into generating robust islet type products for potential clinical application. And here we are, in the developmental phase and trying to work out what we need. But I think all the elements in terms of a first in human trial are already there in terms of developing and growing these cells initially in the bioreactors and being able to expand iPS cells and differentiate them into a safe islet product that could be potentially implanted in a patient are already there. And I think that the challenge now is how do we scale that for more mass manufacture for patients, and how do we think about mass manufacturing this for thousands of patients? So those are the opportunities ahead as we face and replicate these kind of islet-like cells that stain so beautifully with dithazone and behave and reverse diabetes over the course of time. So we've tested these with Pat McDonald in the lab, and on dynamic glucose stimulation, they produce appropriate amounts of human insulin. And here, when the stem cell islets are patch clamped, they have early and late phase exocytosis and behave very similarly to human islets. So we're very excited about being able to take this forward and move forward. And I'm not the one doing any of the work here. We're very lucky to have Nuria Costa Gomez and Ila join us in the lab most recently to work as postdocs and help drive this work forward together with our technicians, Rena and Parveen. Very, very fortunate, and again, emphasize that this is all teamwork. So some of the elements of what we're working on currently is how do we purify these cells to make sure there are no off-target cell elements if we move forward to consider first-in-human trials. And so one of the areas that Nadish has been working on now and is focusing on is how to use both physical methods of disaggregation and reaggregation together with potential a number of different chemical methods, such as this one, to eliminate the risk of any off-target growth during the growth and expansion phase. And as we make progress with that, we're also planning now the GMP grade all the documentation that's required to move to the Alberta cell therapy manufacturing facility that Dr. Greg Corbett runs together with his team, so that we can plan and move forward with the first-in-human trials over the next 12 to 24 months or so. We know that's an ambitious target, but we can short-circuit this, I think, quite significantly by using the same macroencapsulation devices that ViaCyte have used to encapsulate the islets derived from these autologous stem cells, transplant them into patients, remove them at different time points with these Sentinel devices, and see how well they're surviving. And as we move this work forward, we will then move to the deviceless site that Dr. Andrew Pepper and I developed in the lab as a pre-vascularized site for subcutaneous islet implantation. And I think as we generate sufficient safety data, then we'll move from there to being able to put these kind of cells into the liver in full therapeutic dose to reverse the disease. So that's why I'm really excited about this. We'll have to recognize that there are different opportunities with both these universal embryonic stem cell banks as well as the autologous IPS products. My bias, I think, and interest, because I'm a transplant surgeon and recognize the risk of the long-term anti-rejection drugs, being able to develop autologous therapies is particularly exciting, but it's also comes with its own challenges. Mass manufacture is clearly going to be easier with the universal cell product, but they are currently allogeneic. The gene editing approaches may overcome that, but that has to be proven, and we need to wait and see. How mature do these cells need to be when we transplant them? It's difficult to say. Tim showed you how he moved these cells from stage 4 to stage 7 and made them much more mature and function earlier after transplantation in mice, and that might also allow us to move forward more rapidly into patients. The autologous product, because it's patient's own cells, for me is conceptually more attractive, but it's clearly challenging today, and we're trying to work out how we can move forward with that and how we might regulate it. And these are some of the challenges that we face as we move forward. So Tim and I have put together an advanced cell therapy for diabetes concept, ACT4D. It began as a scratch on a piece of paper. We've put a number of grants in now to see if we can move this forward in terms of scale up, looking at the clinical trials, looking at all the legal and ethical frameworks involved, and how we approach this to move forward and look forward to an early future. And Tim and I are very fortunate to work with a number of brilliant investigators, including some of them shown here: Russ Greiner, Pierre Boulanger, Tim Caulfield, Christina Nostro, and many others, Knut Woolgen in Kyoto, to move this forward to treat patients and look at the complexity of this process. So scaling up these cells, automating it, looking at the legal, regulatory, and ethical and economic challenges as we move forward in the next year or 2 or 3 or 4, will take this work, I think, from concept to meaningful clinical trials. And at the same time, I think as a spin-off, will generate a large number of iPS high-quality cells that could be used for other areas of regenerative medicine. So we know that in 2040, Deloitte has predicted that 40% of therapies will be, US healthcare spending will be spent on curative therapies. That's not possible today. And we know there's been massive and exponential improvements. If we step back for a moment and pause, the number of mobile phones that are around today has doubled in the last 48 months. DNA sequencing cost $1 billion just up until recently, 13 years ago, and now cost less than $100 and can be done in less than an hour. So it's quite possible to think beyond the current and look to the horizon of what is possible in terms of generating robotics and biomechanical engineering and machine learning to apply to all of these processes. And we're currently working with a number of different companies, including this one, the Cocoon platform from Lonza, that has conceptualized the idea of making these automated machines for generating personalized medicine, and having a bank of these in what they describe as an orchard to mass manufacture human products at reasonable costs for patients. And we're trying to figure out too how we can automate this process using robotic engineering and a number of other approaches that will make it possible tomorrow what seems impossible and challenging today. We've Tim and I have connected with companies such as Celino, which has a laser capture device for eliminating any off-target cells. This could be very useful as we try to purify the iPS cell products. And other companies such as Tree Frog Therapeutics can mass manufacture stem cells by temporarily encapsulating them in a membrane and then dissolving off the membrane over the course of time. This has led to massive cell expansion in their kind of products. And we're looking to these kind of technologies as we move forward. So I'm certain, and I think many of us are, that diabetes will become a thing of the past if all goes according to plan. The plan is a big one, it's an ambitious one, and it's going to require a lot of effort from many, many people. My young little daughter upstairs, playing upstairs this morning, Abigail, is looking to a brighter future, and we look to our scientists in the future to follow in our path and generate a really bright future for diabetes and for all of us. So thank you for listening. I'm very happy to provide answer some questions looking at cell transplantation from the beginning to the end of insulin. Thank you.
Thank you, Dr. Shapiro, for this presentation. And I believe the donors and the patients are a crucial part of diabetes research. And we are very fortunate to have some of these patients attending this seminar today. And Cheryl, she shared with us her story, and she's 5 years past her third transplantation, and her A1C is once again climbing. So according to her words, the honeymoon is once again fading. And she's very often asked, and she wants to ask you, why some patients need to repeat the islet transplants. And about a cure, although scientists have identified many factors related to diabetes, what exactly is the thinking of how best to eliminate the need for treatment?
Yeah, so there's a lot to unpack in that question. I won't attempt to answer it all, Cheryl. But why does one patient have a islet graft that works 21 years later, and why does another patient like you, the honeymoon period, recur after 5 years or so? It's really hard for us to understand, and because we've been putting these cells in the liver and they're not easy to biopsy, it's hard for us to visualize exactly what's happening in patients. We assume, but don't know, that maybe in some patients they have more active autoimmune system, and that's why we've in some of our trials have been giving more heavy immunosuppression and certainly depleting T cell that we didn't do in the early trials with the islet cell transplant to see if we can mitigate the recurrence of autoimmunity over the course of time. But I think as we study and learn this in more detail, we have to work with human immunologists that can understand this better than we can as scientists and clinicians, to see if we can intervene with all of these pathways, because whether they're islet cells or stem cells, we still need to protect them in the longer term. And maybe these strategies that gene edit the cells and make them more robust and locally shielded could be one avenue. They certainly seem to be promising, but will they really deliver for mass numbers of patients like you? Difficult to say in the longer term until those trials are done. And I'm quite certain that the progress will be made, but it's going to be an iterative process. So it won't all be done in the first go round, most likely.
Thank you. Another question is about the relationship between abdominal obesity and the blood sugar control with islet transplantation. Do you have anything to say about that?
We have patients with different body mass index, and we also have patients with different insulin requirements. We know that a patient that has a low insulin requirements, we can calculate it as we control for the body weight. So we do how much insulin is the patient requiring per kilogram. So somebody who's very insulin sensitive might be using 0.2, 0.3 units per kilogram. Somebody that's resistant to insulin, and they're often more obese patients, could be using 1 unit per kilogram or sometimes more than that. So combinations of type 1 and type 2 diabetes are being seen. Now, presumably we can overcome that by the dosing of the cell product. So today we're limited by the amount of dose we can give, because we basically give to a patient what we get out of a single human pancreatic islet, out of an isolation. But for the future, we could potentially dose patients and titrate what the patient requires, since we'll have a limitless supply of cells, and we can keep adding that, topping up until we reach a point where a patient has a normal reserve. Today, when we put the islet cells in, we give enough to allow a patient to be separated and independent from insulin, but we don't give them a completely normal endocrine engraftment component. So to do that would require many more cells, and we have the ability now with the stem cell products, embryonic or iPS, to deliver on that.
Okay, thank you. I think we have time for a very quick question. Do you have an answer for the previous question about the genetic of a person and the anti-rejection meds required in the Edmonton protocol?
Yeah, I don't think we have any studies in islet or in other solid organ transplantation today that really robustly predicts the efficacy of a particular therapy. But what you could look at, and I think the stem cell opportunity presents itself in this way, because we can make massive numbers of cells, you can test them in the dish. You can test them against different anti-rejection drugs. You can test them for, potentially you can mix them with the serum of patients and carry out mixed like a mixed lymphocyte reaction with the islet, and see which patients respond better and what's needed to really get to an individualized and personal medicine-based therapy that we've never been able to do before. So I think that's on the horizon. So it's an exciting and I think an important one.
Okay. Thank you so much. So I'm going to pass now to Dr. Peter Light for the closing remarks for this event. Thank you.
Wow. Thanks, everyone. A big thank you to our speakers, Alison, Krista, Ray, Tim, and James for enthralling and fascinating talks. As Canadian scientists, we truly punch above our weight, as mentioned earlier. We've made some exceptional discoveries throughout the years, and we continue to make discoveries and lead the world in some of the latest technologies. I'd also like to say thank you as well to our two co-hosts, Theo and Stephanie, for taking some of the burden off myself, but actually doing a fantastic job at keeping this symposium going. So thank you guys for that. Regarding the future of diabetes research, I think, we're using the word future quite a lot, but I think the future is almost in the present. I really feel as though a cure is just around the corner. And I'm confident that with the exceptional scientists we have in Canada and right here in Edmonton, that we can be leading the way towards a cure for many forms of diabetes, certainly type 1, but ultimately down the road for type 2 as well. So it's been a fantastic event. We're celebrating 100 years of insulin. We've got an idea of the history for the discovery of insulin and what's been done in Canada and right here in Edmonton since then. It's been, since the discovery of insulin, it's been 79 years before the publication of the Edmonton Protocol. I'm hoping it will be a lot sooner than 79 years before we can actually say that we have an effective cure for diabetes. So with that, I thank you all again. I thank all the participants and the attendees for this symposium. It's been a great event, and just to let you know, it has been recorded. So it will be on our website, adi.ualberta.ca, in the next week or so. So if you missed anything or you want to listen to some of the talks again, please check that out. So again, thanks everyone. It's been great. And with that, if there are no further comments, I'll end the symposium. Thank you, everyone.